Good morning, dear friends. Good morning, one and all. Myself, Dr. S.R. Deshmukh, SK Gandhi College, Kada, would like to welcome you, one and all. It gives me a great pleasure to extend you all a very warm welcome on behalf of Department of Botany, who has clubbed all of us for this national webinar. Department of Botany is established since commencement of our college, that is since 1996. We have in our institution both UG and PG courses run by Botany department. Encouragement, enthusiasm, excellency is one of the major aspects of the organization with an object of encouraging the spirit of research and innovations. Department of Botany feels proud to host this national webinar. This event is not just a single day routine seminar. Rather than it is a composition of 5 eminent speakers lectures which will enlighten which will provide enlightenment by each specialized in specific knowledge areas. Once again, wholeheartedly, I welcome you one and all on behalf of Department of Botany, SK Gandhi College, Kada. Honorable dignitaries, excellencies, learned desks, respected teachers, a very good morning to one and all once again. I, Dr. Suparna Deshmukh, feel privileged to welcome you all on behalf of organizing committee of this national webinar on role of biofertilizers for sustainable development, organized by Department of Botany, SK Gandhi Arts, PH Gandhi Commerce, and Amulok Science College, Kada. Well, Albert Einstein said, if we knew what it was we were doing, it would not be called research, would it? It is on the occasion like this we get opportunities to test our knowledge and our understanding. We look forward to get an exposure about what the best of the brains think about this very dynamic issue of and alarming issue of role of biofertilizers for sustainable development. It is our customary in our Indian culture to commence any event by lighting lamp and by praying Almighty for this success. Accordingly, I would like to welcome our Honorable Principal, SK Gandhi College, Kada, Dr. N.S. Rathi, sir, Honorable Vice Principal, Dr. J.M. Bhandari, sir, to come forward and kindly light the lamp for commencing our webinar today. Sir, please. Along with Rathi, sir, Principal, Gandhi College, Kada, Vice Principal, Dr. J.M. Bhandari, sir, we have a special guest, Principal Nilesh Jadhav, sir, Pharmacy College, Kada, Sri Amolak Jain Vidya Prasarak Mandal, Kada. Deepam Jyoti Parabrahmam, Deepam Sarvatamopham, Deepe na sadhyate sarvam, Prathamam Deepam Namostute. We are praying Almighty Lord for the grand success of today's webinar. Dr. Principal Dr. Rathi, sir, Principal Dr. Nilesh Jadhav, sir, Vice Principal Dr. J.M. Bhandari, sir, Head of the Department Botany, Dr. Yunus Sayyed, sir, Dr. S.S. Patade, sir, are lighting the lamp for praying the enlightenment to the Lord. A formal inauguration will be announced by auspicious hands of our principal, Dr. N.S. Rathi sir. May I now request Dr. Rathi sir to just announce the inaugural function, just announce the formal inaugural function of our webinar. Dr. N.S. Rathi sir, please.
Good morning to one and all. I, being a head of the institution, I welcome all the dignitaries, especially the invitees, keynote address, Professor C. Manohar Chari sir from Hyderabad, and Professor A.B. Ade sir from Pune, Dr. B.D. Takate Nevase, and Professor Umesh Mogle from Jalna. I declare here the national webinar has been inaugurated, and today's webinar's theme is role of biofertilizer for sustainable agriculture development. This is a very good topic, and I hope that during the one-day national webinar, all the participants, they will take the benefit of the concerned speaker. So once again, I welcome all the dignitaries and resource person with this, and again I declare that this webinar has been inaugurated. Thank you. Thank you very much.
Thank you so much, Principal Dr. N.S. Rathi sir. Thank you for your best wishes for this webinar. May I now request Dr. S.S. Patale sir to come forward to give the introduction about this webinar. Dr. S.S. Patale sir, please.
Good morning. Welcome all the participants in today's one-day national webinar. Today's chief guest and innovator of webinar, Professor C. Manohar Chari sir, Hyderabad. Speaker, Professor A.B. Ade sir, Head Department of Botany, Savitribai Phule, Pune University, Pune. Then second speaker, Dr. B.D. Takte sir, Nevasa. And third speaker, Professor Umesh Mogle from Jalna. Honorable Sri Kantilalji Chanudia, President Sri Amolok Jain Vidya Prasarak Mandal Kada, Honorable Sri Gokuldas Ji Meher Sahib, Honorable Sri Yogesh Kumar Ji Bhandari Sahib, Honorable Sri Hemant Kumar Ji Pokarna Sahib, Secretary Sri Amolok Jain Vidya Prasarak Mandal Kada, Honorable Dr. Umesh Kumar Ji Gandhi Sahib, Treasurer and Coordinator Sri Amolok Jain Vidya Prasarak Mandal Kada, all the trustee Sri Amulok Jain Vidya Prasarak Mandal Kada give us permission for organizing this webinar. Our dynamic principal, Dr. N.S. Ratti sir, Vice Principal, Dr. J.M. Bhandari sir, my department head, Mr. Syed I.G. sir, who is the convener of this webinar, all the members of organizing committee of this webinar, all my staff, colleagues, non-teaching staff, students, researchers, teachers, and my friends who were joined this webinar from various parts of the country. It is great pleasure to organize the national level webinar on role of biofertilizers for sustainable agriculture development. It provides a platform for the budding researchers and younger generations. Today's webinar, more than 250 participants were registered for this webinar. The webinar is aimed to promoting discussion, information, sharing knowledge, and to have new insights in recent development for biofertilizers. A biofertilizers means it is a substance which contains living microorganisms which, when applied to seeds, plant surface, or soil, colonize the rhizosphere or the interior of the plant and promotes growth by increasing the supply or availability of primary nutrients to the host plants. Symbiotic nitrogen fixation, that is rhizobium species. Asymbiotic nitrogen fixation, that is azotobacter, azospirillum, etc. And phosphate solubilizing bacteria. Then trichoderma, glyocladium, aspergillus species, and many microorganisms were used for the preparation of biofertilizers. The theme of the webinar is role and types of biofertilizer in agriculture. Role of microorganisms for preparation of biofertilizer. Microbiology and fungal biotechnology. Use of biopesticides and biofungicides. Ecology, environment, and agricultural development. Friends, our botany department is one of the leading departments in our college, UG and PG departments. The faculty of department is actively engaged in research activity. The faculty has published more than 100 research papers in national and international journals. The department faculty completed 5 minor research projects and 1 PhD students were awarded and 2 are working their PhD. And the department were organized 3 national conferences and 1 workshop. Friends, role of biofertilizer in agriculture is very important for the control of pollution, improvement of crop productivity, and safety to the humans and all living things. Friends, the webinar is organized for students, research fellow, teachers, farmers, so as to provide a common knowledge and experiences, innovations, and information. I am very much thankful to all the participants who were joined this webinar. Thank you, one and all.
Thank you so much, sir. Thank you for your nice introduction. Now, let us have the pleasure of listening to the opening address of Honorable Professor Sri Manoharachari from Hyderabad. May I now request Manoharachari sir to kindly enlighten us by your nice words. So it will be my pleasure to invite Manoharachari sir to deliver the keynote address. Sir, please.
Hello? Hello? Hello?
Sir, you are audible, sir. Please. Yes,
Okay,
yes, sir,
okay.
you are audible. You are
Okay,
audible.
thank you.
Welcome, sir.
Dr. Patale?
Yes, sir. Yes, sir. Yes, he is here, sir. Patal sir is here. Yes, sir.
Yeah. Dr. Patale?
I'm not able to share the screen.
I'm not able to
Yes, sir. Yes,
share
sir.
the
Yes, sir.
screen. I'm
Okay,
not
sir.
able to share the screen.
Within a minute, we are making you host, sir. Just a minute. Just a minute.
Okay, okay.
Yes, sir. We have made you host. You can share your screen now, sir. Sir, please. Sir, would you be able to share your screen now?
Yeah. But screen I want.
Yes, sir. We have made you host, sir. You can share your screen now, sir. You can share your screen.
Okay, right.
మీరు ఇందులో మాట్లాడండి.
Hello?
Sir, just a minute. I would like to invite before your lecture, Sayyed IG, the head department of botany, SK Gandhi College, Kada, just to give the introduction. Sir, please.
ఇంకెవరిని పిలిచింది మధ్య.
Sir, you have started your screen sharing. We are able to see the shared screen.
Okay, good.
May I now request Sayyed IG, sir, to come just give the introduction.
Okay, good. Thank you.
Thank you, sir.
It is my pleasure to introduce the chief guest and inaugurator of national webinar or role of biofertilizer in sustainable agricultural development, Professor Sri Manoharacharya from Usmania University, Hyderabad. He is the dedicated educationist and researchers vast experience of administration and research. He is also worked as a vice chancellor of Oriental University, Indore. He was the dean of science faculty in Usmania University. His teaching experience is more than 4 decades. He was mature professor of CSIR. He is also enjoyed post-doctoral research for Horizon and Horizon University. He had made 6 countries for the publication of research paper in reputed Indian as well as foreign journals. There are 30 books to his credit. He is member of various prestigious national committees of Government of India. He is a chairman for UGC and Development Affairs. He has visited Thailand, Hungary, Netherlands, and China for international conference. Apart from this, he awarded various awards for his devotion in the field of teaching and research. He has discovered and described 20 new genera and 82 new species in fungi. His contribution in biodiversity, taxonomy, conservation,
fungi. Presently, he is serving as a NASI senior scientist. Sir, thank you very much for being with us. Thank you, sir.
Thank you, sir. Thank you for your nice introduction. May I now request Manora Chari sir to enlighten us by your
Hello,
hello, sir.
audible.
Yes, sir. Yes, sir. You are audible.
Thank you.
Very loudly audible.
మీరు మాట్లాడండి.
Yeah, now, thank you very much. And for the nice introduction, Dr. Sayed sir, for having given me an opportunity. At the outset, I thank respected Honorable Principal, Dr. Rathi, and Vice Principal, Dr. Bhandari, Head of the Department, Dr. Sayed, and the Organizing Secretary, Dr. Patale, and of course, another speaker, my beloved friend, Dr. Professor Ade from University of Pune, Department of Botany, and all other delegates and all other speakers. Now, I am going to speak about role of microbes and fungi in plant diseases and pest management. The reason that I have chosen this particular one is that the role of biofertilizers, though it is important, but the most important thing is the control of the diseases. Biofertilizers play an important role in the plant growth and as well as sustainable agriculture. And the problem being that the biofertilizer technology with reference to mycorrhizal fungi has become very difficult because mycorrhizal fungi could not be multiplied except the ectomycorrhizal fungi, whereas arbuscular mycorrhizal fungi, being obligate symbionts, cannot be multiplied under the conditions which are available as on today. Commercialization is very difficult. But nevertheless, I would like to say that the sustainable agriculture with reference to India is very important. No country can progress without education and agriculture. That is the reason why the topic chosen by the college authorities and Dr. Patale is very appropriate and relevant to this particular country and particular to Maharashtra also. Now, as far as the biofertilizer technology is concerned, why it is important? Because India is one of the leading countries which have produced 300 million tons of cereals and 300 million tons of commercial, that is the commodities of horticultural crops. It is a leading country as far as the horticultural crops is concerned, but nevertheless, our population being very high, we cannot supply all such kind of cereals or whatever it is that is necessary for the growing population. So in order to achieve the food security, we have to emphasize more on plant diseases, their management and plant growth and the crop productivity. So by 2030 or 2040, India is going to be #1 in population. Though we say that biotechnologically we have become forward, agriculturally we are also forward, but nevertheless, but the most important thing is that we are very much forward in the production of the population. So we need to have more of food grains for the growing population, and in the years to come, we have to achieve this. And in this particular one, I must emphasize the role of green revolution and Nobel laureates. You know that under Dr. Swaminathan, the things have become very easy for us to achieve green revolution, second green revolution. But now, is it necessary that after first green revolution, is it necessary that we need a second green revolution? So second green revolution has become necessary because of the fact that the fertilizers which are used by the farmers have been non-judicious, and therefore soil and plant health is getting deteriorated. The pesticides and fungicides which are used in larger quantities without any scientific basis is also spoiling not only farmers' health, but also the health of the crop, and as well as the useful microbes which are there underneath the soil, they are getting destroyed. And the one more important thing is that climate change is going to be a major setback for all our agriculture, and not only in India but also elsewhere. So, keeping all these things in view, we need to have a second green revolution also, and also further development of the crop yields and as well as the forest establishment. Because I'm talking about forest also because of the fact that against 33% of forest, we have only 22%. 11% of forest has been lost due to the man-made activities and selfishness and jealousies of the people who have spoiled the forest for their own purpose and for their own business. So now this is particularly, I would like to emphasize that plant diseases are mainly caused by bacteria, viruses, mycoplasma, fungi, insects, and biotic and abiotic states, and weeds take a lot of that is all our crops also getting destroyed by the weeds also. So our agriculture production is getting reduced to 40% level by means of plant diseases, as I mentioned just now only. So on average, it may be 25 to 32% of crop yield is reduced, but resulting in 1,50,000 crores of our economy is lost in our country. In other words, we are losing many of the crops because of all these organisms. So whether you use biofertilizers, whether you use fungicides, whether you use pesticides, whether you use any biotechnology, whether you use any kind of technology, it is most important thing is the plant and soil health has to be maintained. I think Patale was mentioning about rhizosphere. Really, it is very important that rhizosphere is a specialized ecological niche, and many of the useful microorganisms are there around the root, and we have to see that all those organisms are brought into culture. Unfortunately, whether it is bacteria or fungi, useful bacteria or whatever it is, we could not culture them. Only 7% of fungi and bacteria and actinomycetes have been cultured so far in the whole of the world, and also including in India. That means we don't have cultures to boost the biofertilizer technology. So in order to achieve sustainable agriculture, we need to have the mass multiplication of all these biofertilizer organisms, the PGPR and as well as Frankia, which is also nitrogen fixing associated with that of Casuarina, and also some of the fungi and phosphorus solubilizing fungi and mycorrhizal fungi and so on and so forth. So all these organisms have to be cultured and maintained in the form of commercial basis, just like any other fertilizer that is sold in the market. If that is not possible, then sustainable agriculture is very difficult. But one important thing that our agriculture scientists have achieved is that plant breeding programs have been very successful in India, and a lot of disease resistant varieties have been produced in India, and they are now used in the crop productivity, and they are helping us in the sustainable agriculture. So we need to look into all these aspects. So in spite of severe criticism on the use of fungicides and pesticides, and farmers, specifically from that of Maharashtra, they recognized that usage of these fertilizers in larger quantities and fungicides and pesticides will give them good yield. So though there is the health setback to the farmer, and as well as soil health is also lost, but still everybody is interested in producing more and in getting more of money, and that is a major issue in our country. So the issues projected are in the case of pesticides and fungicides, is the issues that are projected are residual effect with reference to pesticides and fungicides, and then resistance to pollution, and then contamination, soil contamination, and all such kind of soils need to be bioremediated. They're very difficult task in our country. And you know, in Haryana and Punjab, many of the soils have become sodic because the non-judicious application of fertilizers and fungicides and as well as pesticides. So the soils got contaminated, and thousands of hectares of land is now waste, has become wasteland. So bioremediation of such kind of land is a very difficult task for us. And spurious sale, that is, you know that many of the pesticides and fungicides, and also some to some extent fertilizers also, they are using, they have become spurious because they mix with some other things. So now the importance is laid down by the government of India, on organic farming, where you can see the manure, cow dung and so on and so forth, which can be used for the product sustainable agriculture, which is also a part of biofertilizer technology, because it has been an old practice in our country that such kind of organic manuring has helped us to boost our crop productivity. Probably there is a need to identify correct and lethal crop specificity and also implementation of the PPP mode. That is very important. Now, this is the crop diseases can be controlled by crop rotation, that is the lower inoculum potentiality, adding the amendments like antagonistic, like biocontrol agents and stimulators to some of the PGPR and other such kind of stimulators, and then pH adjustment and tillage, modify the soil structure, and planting of the date of selection, and organic amendments and irrigation practices, and using of trap plants for mycorrhiza, and adding of antagonists like antagonistic bacteria and trichoderma and so on and so forth. Now, just as people would do not take care of themselves and are more susceptible to diseases, if you don't take care of the plants like crops, then naturally they get disease. So plants can also be more vulnerable to infection when their nutrition is become optimal. So that is very important from the point of nutrition is also important, and whatever that is important, that has to be taken into consideration. Over dependence on pesticides and fungicides is hazardous to man and environment. Now we know that why diseases have become important. Late blight of potato in 1845 made the Ireland people to suffer a lot. Economy is lost. People have moved from Ireland to other countries. Coffee rust in Sri Lanka in 1875 made the people to forget about the coffee drink, and now they are adapted to the tea plantations, and there is no such kind of coffee that, though it is available, it is a small quantity, but earlier it was sending all its coffee to other countries also and exporting that. But Bengal famine in India, because of Elenthosorium variety in 1942, has shaken the whole of the Bengal and as well as other parts of the country. And ricey blast disease has become very important in South India, where people have suffered a lot. So what I want to tell you is pesticides and fungicides are poisonous entities and hazardous to man and environment, and they have to be used very judiciously. Scarcity of food to lead to the development of fungicides and fungicidal resistance is another important problem. So these are some of the fungicides which are commonly used in our country, and say the year of inception, that is their use, I started from so and so here. I don't want to go into the details, but you can just read the table just like that only. Now this is these are the biological control methods. These you can see the most important thing in the case of biological control is antagonism. You can see here that the fungus is getting this is one actinomycete in the middle plate. You can see that is controlling the pathogenic fusarium. The other one is right side is the trichoderma, which is now commercialized and produced all over the world. And in India also commercialized trichoderma is available as a biocontrol agent. Now you can see that in the case of the lower one, you can see that last plate, this one fungus is inhibiting the other pathogen. So these are some of the important antagonists where you can see antibiosis is the basic principle that is taking place in this particular one. So based on the antibiosis and antagonism, the biological control has been framed, and now many of the biological control agents are now available in the market in the form of commercialized products, which will help in the control of the diseases and as well as sustainable agriculture. You can see here, these are the selected examples of root diseases controlled by application of antagonistic bacteria. And now very important one is that seed priming, that is putting the inoculum of antagonists on the seed, and seed coating, that is called. Now recent term is seed priming. That has become very important method of applying all these biocontrol agents. Just roll them in a particular type of antagonist like a trichoderma, PGPR, or any other thing, then which will control the diseases or which will promote as a biofertilizer like PGPR, then it will help, and just plant them and then you will get very healthy plants. So these are some of the antagonistic organisms that I have given. Even Aspergillus niger is also antagonistic fungus towards Fusarium solani. It's the most common fungus which is culturable, and one can use all such kind of fungi and always bacteria and trichoderma and so on and so forth. For example, penicillium is age-old fungus which is known for antibiotic production. Many penicillia are also antagonistic and biocontrol agents. And Pacillomyces, which recently it has been patented by Dr. P.K. Chakravarti, the IPS president. And these are the principles of biological control. It is the process which reduces or controls the pest level or disease level by using another microorganism. That is the definition. So use of antagonistic microorganism, whether it's bacteria or fungus or actinomyces, it is that microorganism that antagonistic to the pathogen. And then the microorganism will destroy and inhibit the growth of the pathogen. And it is the eco-friendly method of applying the biological control method. So these are the antagonists which are used in the case. Already I have shown you the plates where petri dishes I have shown some of the fungi. So the basic principles are antibiosis, competition, commensalism, and as well as parasitism. That is predation, overgrowth of the hyperparasites, overgrowth of a biocontrol agent, overgrowth, that is called predation or parasitism. Commensalism, antagonist makes the environment unsuitable by producing some of the metabolites or secretions. Competition, it is competition between the two organisms, the pathogen and as well as the antagonist. And antibiosis, inhibition of the pathogen. Now these are some of the, See, PGPR has been used as a, these are some of the potential biocontrol agents used in the biological control of plant diseases. So these are some of the things which are available in the market. And these are the fungi, Chaetomium globosum, which is a common, actually it is used in textile industry, and it produces tons and tons of cellulase, the biotechnology product, which is used in textile industry and also in the ornaments. The stones that you use, in the case of diamonds and other things, they get polished by this particular cellulase enzyme, very important fungus. And chitinase enzyme, which is produced by many of these fungi, is also used as a biocontrol agent. So these are some of the common potential biocontrol agents, and they are available in the form of various products that I will show you later, and that is available in the So the trichoderma is a biocontrol agent. You can see nature is armed with the teeth, and the challenge is to manipulate all such kind of organisms in a beneficial way at a molecular level, and by means of powerful manipulations will help us, whether it is by traditional method or molecular method, advanced methods, you can insert that particular genetic material into biocontrol agent. If you know that there is a resistant gene which will control the disease, and that resistant gene from trichoderma can be taken out and can be introduced, it can be mutated, and then it can be it will become a potential organism. So you can see a mutant resistant trichoderma species, and we have actually by introducing the gene, we could make it out, and by exposing this particular UV light, and these isolates suppresses a lot of soil-borne control, they will control a lot of soil-borne diseases, very important, and we can spray all these things even through helicopters and other things also, that is possible. So this is trichoderma is used as a very good biocontrol agent, and many biopesticides have been proved successful in a large number, including the case of field site, and also the fruit and flowering crops and other things also will be helpful for us. So trichoderma strains, they are able to control number of phytopathogens. I have reported around 13 trichoderma species from India, and we have brought out a small booklet also, which is also made available. I have also published one book on monograph on soil fungi. This is also available in the market, IK publication from New Delhi, and where we have mentioned about many of the trichoderma species which are occurring in India can be multiplied and are suitable. Even in cow dung you can multiply. Even in the case of sawdust you can multiply. You can also fly ash also you can multiply. You can also baggage also you can multiply this one, and you can prepare a very good biocontrol agent. Leave alone the sophisticated company products, but we can also ensure this kind of technology even through rural technology also, which is possible. So various strategies have been explored to control these pathogens, and this reveals the importance of biological control agents. These are just some of the commercial formulations of trichoderma that are available. You can see BNAP T, and you can see that Trichodex, which is, they are all available in India. So Root Pro, which will also see, trichoderma is not only a biocontrol agent, but also is a growth promoter, like PGPR, plant growth promoting rhizobacteria, Pseudomonas. It is also a growth promoter. So for sustainable agriculture, trichoderma is not only useful for control of the diseases, but on the other side, it also produces lot of growth promoting substances. And of course, you know, Gibberella piscicorae also produces gibberellic acid, which is also a potential growth promoter. In the case of Maharashtra and other places, seedless grape varieties have been produced by spraying this particular gibberellic acid. So we have such kind of 100 and odd products, biological control agents available in the form of commercial ones. And you can see PGPR very easy to isolate from that of the rhizosphere region. And it has got multiple effects, not only as a biological control, but also it is a growth promoter. Many of the species are available in India. Some of them are culturable. And Pseudomonas fluorescens, they help in the nutrient uptake and multiplication of the plant roots. And also the hormonal secretions also is possible. And you can see siderophores are produced by this particular, even trichoderma also produces siderophores. Recently, I have brought out trichoderma in agriculture and beyond agriculture, published, I edited that volume along with Dr. H.B. Singh and Professor Ajit Verma. And recently, just 1 month back on it has come from the Springer, and now it is available through Amazon also. And very good research papers are available in that particular one about the trichoderma. So this is PGPR plays an important role for sustainable agriculture along with the trichoderma. So now some PGPR, which will also be used as biocontrol agents and also plant growth promoters. You can see Pseudomonas fluorescens is the major one, where it can control the damping of disease and root rot, but also it is a growth promoter. It is also biofertilizer. And you can see number of strains of Pseudomonas fluorescens are available. Bacillus subtilis is also there. And also you can see Bacillus cereus, and you can see Pseudomonas putida. But for Pseudomonas putida, you have to conduct the test, taxonomic test, because some of the strains are also allergic, and that has to be established before the use. So PGPR is helpful not only as a growth promoter for sustainable agriculture, but also for biocontrol agent. Now mycorrhiza. See, altogether 640 research papers, out of which 60 that we have published only on mycorrhiza. And 3 new species of mycorrhiza also I have published, and around 82 new species of fungi and 220 new genera I have published. And some of the mycorrhizal species I have tested on ectomycorrhizal and as well as the arbuscular mycorrhizal fungi I have tested even on the pinus and as well as plantations, and also on these plants. You can see chickpea, and then the tomato, and then cucumber, and then pinus and mung bean. And it has been very good, and that it not only it controls the diseases but also enhances the plant growth. It is also a biofertilizer technology. It is used in biofertilizer technology, but only difficulty with arbuscular mycorrhizal fungi, though they say that some of the capsules are available, some commercialized products are available, but it is an obligate symbiont. It is very difficult to multiply this, and therefore one has to go for its multiplication under pot culture only. That is Madam Masse, she has proposed from England. Still it is used in many of the countries, and including our country, we also use the same technology, but for multiplication, it is very difficult. But commercial products are available in United States and others. For example, for $52, there are 10 tablets which are sold in United States, in Canada also, and so on and so forth. So mycorrhizal tablets are available in other countries, and India needs to have that kind of technology to be adopted, and somebody has to take up this particular technology for sustainable agriculture, not only as a biocontrol agent, but also as a biofertilizer. So now myco-insecticide, entomopathogenic fungi are called myco-insecticide, and in nature they are there. These are some of the species which are there. Bavaria, you know, Professor Niranjana from that of Mysore University, from biotechnology department, he has used the berry, the coffee berry, which is where the insect borer is there. It got controlled by spraying this particular Bavaria bassiana. So we have such kind of technology also. We have isolated all these fungi from insects alone, and we have used them against insects only. So entomopathogenic fungi can also control number of diseases. These are some of the insects wherein you can see colonized by the fungi. And this is, you can see Numeria, and also in the case of Phaceolomyces, so on and so forth. And Metarhizium, which is also controls the mosquito larvae production. That is also useful. Coelomyces, a fungus which controls mosquito. And these are all some of the commercially useful bioactive products derived from entomopathogen fungi. Dr. M.V. Deshpande from Pune, that is a national chemical laboratory. He was there earlier working there, but now he has got his own company where he is able to manufacture some of these commercialized products, and that is which are useful to control number of insect pests. And some of the plants also very useful for the control of many of the plant pathogenic fungi. Your Vinca rosea, Catharanthus roseus, is a very good, not only anti-cancer plant, but also its alkaloid is useful to control a number of diseases. And like that, we have a number of plants I have shown you. Prosopis uliifera, which is commonly known as a weed. From that also we can path. And you know, Feltophorum is also a plant, big plant, and you can use all these plants to control number of diseases. We have got, now we are screening all such kind of botanicals, the chemical that is responsible for the control of the plant diseases, and around 122 plants we have screened so far. And now we have isolated only 2 compounds from that of Catharanthus roseus and also Peltophorum, which we have given for DBT for patenting. And already we have patented, and we are looking for the productivity in the form of commercial one. So this is botanicals. Why it is not coming?
Excuse me, sir.
Just 1 minute, 1 minute.
Yes, yes.
It is not able to come, some problem.
Sir, Yes, sir. We can see your screen, sir. Very nicely. We can see your presentation on our screen, sir.
Okay, okay. So botanicals on plant diseases, and that is there. 1 minute, just 1 second.
Yes, sir. You can take your own time, please.
కళ్యాణి మాటి పోయింది అది వస్తలేదు. Last, I think there are only 2 more slides, madam. Just wait, 2 more slides, I think.
Yes, sir. Yes, sir. You can take your own time, sir, please.
ఉన్నారు కదా ఇక్కడే.
ఇది ఇక్కడ రావట్లేదు స్లైడ్. ఓకే, వస్తలేదు కదా. Okay, some problem, I think it got stuck. And now another technology that I try to tell you is the nanotechnology. The next one. Yeah, this is nanotechnology that is used in the case of productivity and also in the case of disease control. Nanoparticles of plants, microbes, and fungi, they have been isolated. And you know, Ahmad from that of NCR Pune, now he is in Aligarh Muslim University, has worked on that. And also Dr. M.K. Rai from that of Amravati University has isolated nanoparticles from that of FOMA. And now he wants to apply for the control of the diseases. And bacterial spot of tomato caused by Xanthomonas has been controlled by DNA-directed silver nanoparticle grown, and of course on Graphex, Grapheme oxide. And we have got such kind of compounds about the nanotechnology. And it is a recently developed science. And if anybody wants to use it in the field, it takes a little bit more time, because until and unless you establish the nanoparticle size and its application under laboratory conditions and as well as field condition, one should not go, because it may also take many other microbes get spoiled also by applying in larger quantities of this nanoparticle technology. So there is and fungi in weed control. Weeds are apparently have all been a nuisance for the mankind. And we have a number of weeds. I don't want to go into the details, but we have one fungus which we have got, that is Puccinia chondrulina, and which is parasitic on Parthenium bisterophorus. And from Tirupati, we have got Puccinia abrupta variety Parthenicola. We allow by using this particular one, we could control Parthenium in Tirupati. Only by you have to spray the spores, because Puccinia cannot be cultured. It is a parasitic fungus. And alternary alternata is on water hyacinth, and Sarcosporum redomi has been established in UK as a biocontrol agent. So now biopesticides, already I told you, microbes, botanicals, and microbial metabolites also have become very important. And even microbial metabolites also can be converted into liquid and a commercial form and can be sprayed. And see, chitosan is the one which is a fungal cell wall material. I think this is very important technology which has become recently. Chitosan polymer and its oligomers suppress the growth of many of the plant pathogenic fungi. It has got antifungal activity. We have isolated this chitosan from that of Aspergillus niger, and also from that of Bacillomyces, and also from that of Gaunomyces, and also from Alternaria. Now we are in the process of crystallizing this particular chitosan from that of these are the ones. And you know, novel approaches or technologies are many, but clear understanding of the events are very important, including molecular events are very important, and evaluation of the disease, post response, and control of plant diseases. In order to understand all these things, you have to, some technological advancement is necessary. So further, the omic sciences have become very important. They have become integral part of plant pathogen interaction, elucidation of plant pathogen biocontrol agent, and biocontrol agent also is driven molecule generation and expression also, and management of the diseases. Now we have got biosurfactants as a biocontrol agent. There is a one bacterium RF, you know it, Bacillus amyloliquefaciens. It is a hydrophobic bacterium, and it is biosurfactant includes glycolipids, and this is also used as an antifungal agent against many of these diseases, including botrytis cinerea and also Macrofauna fasciolana. So this is a common bacterium which is present in India. So climate change is another important thing which will affect the disease, and maybe they say that of course it is not astronomy to say, nor astrology, but only I would like to say that climate change is going to affect the many of these diseases on cereals in the years to come. But anyhow, let us not bother about that, because there was a lot of glaciation, continental drift was there, many diseases have disappeared, many new diseases have come. So climate change may have a bad effect, but also will also have very good effect also. Some good things also may come because of the climate change. And this is, it is in the door front, everybody is after that, working on that. But I don't want to say much on that based upon the artificial experimentation, because nature is different. Our results in the laboratory, if it is 100%, when you go into the field, it will be 50% only. So I don't want to comment on this particular one. Molecular approaches in the case of plant disease, chemical control is very important. Toxicity has to be established, and fungicide resistance has to be worked out. Deployment of biocontrol agent is important. Usage of resistant cultivar is still considered as mostly dependent method, and potential molecular strategies will help. What are those potential molecular strategies? Resistant gene mediated resistance, cloning and characterization of resistant genes, development of resistant genes through genetic engineering, engineering resistance using the antimicrobial proteins, and plant and non-plant origin, RNA strategies, and RNA silencing also is used, and transgenic, that is also on the cards. Now introduced plant pathogens, some of them plant pathogens have been introduced into India, and our quarantine regulations have to be very rigid. And biosecurity is an important method, is very important for our country, because many people are coming and going, and many regional stations are also established, and international airports are also established, but still diseases are coming. And coffee rust was not ours, but it has come. And late platter potato was not ours, it has come to India. And then crown gall of disease not ours, but it has come to India. So like that, from Xanthomonas vesictorium, this has come from other countries only. So UG99 is now in Nigeria. At any time it can come to our country. And if UG99 comes, it is a rust disease. When it comes to India, our wheat plant will get devastated. And there is also the pyroglaria that is occurring on wheat plant in Bangladesh. It is our doorstep. And we don't have any control over the people movement. At any time, it can come onto our wheat plant also. So there are 75 pathogens which are laid there like that. And these are some of the plant pathogens just I wanted to show you. And then soil solarization is very important physical method. And you have to cover a lot of soil and then disinfect the soil by heat generated from trapping the solar energy, elevating the soil temperature, and that will control a number of diseases also. Thank you very much. This is the university which has given me the bread and butter. I thank my university also, my students and my teachers, and also all of you, and your principal, vice principal, and head of the department, and Dr. Patale for having given me an opportunity to talk to you about this particular one. I never thought that, of course, I was not knowing about the title of the thesis, the title of the webinar, and otherwise I could have spoken about mycorrhiza also. But nevertheless, just like that, I selected that particular topic because I was on the way to some other place, so I could not look into all those things. But anyhow, I think I touched upon biofertilizer technology and biocontrol agent and importance of agriculture, and let us make our country self-sufficient in the crop productivity, and let us see that nobody will be suffer with hunger, and that is the main motto of any agriculture, and any scientist. And agriculture and education, if they are not supported by any country, I think country will not progress much. So anything can wait, but not agriculture. That is what the Jawaharlal Nehru said. Everybody is after agriculture, but agriculture has to be given its own due position and importance. Once again, I thank every one of you and Professor Ade also for having given me an opportunity.
Namaste.
And Principal
Thank you
Vice
so much, sir. Thank you so much
Thank
for
you.
your eye opener talk, sir. Sir, you have explained very nicely the importance of bioremediation of land, sir. You have made us aware about many more new microorganisms and biota available to us naturally. Then plant breeding program of India and the disease resisting cultures developed by India, you have made us aware about it. Thank you once again, sir. Sir,
Thank you.
before expressing my deep sense of gratitude, I would like just to take some few questions available over here. Professor Manoj Kuchekar, he is asking, sir, that you have mentioned PCNB in your slide as fungicide, but it also blocks apoplastic pathway of sucrose or food transport in plant. Then why government has approved that fungicide? Because it also affect on plant growth as well the productivity. Sir, we would like to get answer from you, sir. Please.
Yeah, they have approved because of the fact that in ICR and IRI laboratories, for once they get any fungicide, and first to implement that particular one, they will ask ICR organization to conduct field tests, and after conducting the field tests only, then they will give you a certificate. So they might have done it, and I don't know, but exactly. But nevertheless, I think if Patale sends me that that information, I will ask whenever I go to ICR, I will ask them to retest that particular one and see that something good comes out of that. And if there is a lot of effect on the crop plant and otherwise also, then I think one should look into that. Really, it is something good. I appreciate.
Thank you, sir. Thank you so much. I take this opportunity to extend my gratitude towards you, sir, on behalf of all the trustees, Amulak Jain Vidya Prasarak Mandal Kada, as well as on behalf of our motivator, our respected principal, Dr. N.S. Rathi, sir, as well as on behalf of Department of Botany, S.K. Gandhi College, Kada. Once again, so much thanks to you, sir. You have explained very nicely the use of fungi for producing antagonistic microorganisms, antibiotics, and the biological control. Also, you have explained very nicely how the trichoderma can be used as the biocontrolling agent. Once again, thank you so much for your eye-opener talk, sir.
Thank you, madam. Thank you, one and all. Dr. Ade, how are you? Dr. Ade?
I'm fine, sir.
Doing well.
Yeah, sir.
क्या हो गया? देखो आप हेडशिप लेते ही पैंडेमिक आ गया क्या?
Yes, sir. क्या है अभी वो डेस्टिनी है.
So how is your lab? Everything is okay?
Yes, sir. Yes, sir. It's
Okay. All of you should take care. Maharashtra is having a lot of cases.
Yeah, sir. Yeah.
So wish you good luck. Thank you.
Okay. Thank you, sir. Thank you.
Thank you so much, sir. Today, yes, sir. Today we have invited some eminent speakers over here to enrich us with important information through our discussion on a buzzword, that is the sustainable development. We have with us for our next discussion Professor A.B. Ade sir from Pune. May I now request Zadwar sir, Professor Prakash Zadwar, to give introduction of Ade sir. Sir, please.
Thank you, madam, for this another session of this national webinar on role of biofertilizers for sustainable agricultural development we have another eminent scientist with us, that is Professor Avinash Adesar. It is my great pleasure to say a few words about my teacher. I was Sir's MSc student. Professor Ade has now working as professor and head at Department of Botany at Savitribai Phule University at Pune. He has completed MSc from Amravati University and he is a gold medalist of his batch. And also completed PhD from Dr. Babasaheb Ambedkar Marathwada University, Aurangabad. Sir has total teaching experience of 23 years and also research work experience of 23 years and guided 11 students for PhD for their degree, and now 6 students are working with his able guidance. Sir also awarded or guided the MPhil students. The 5 MPhil students have also completed their degree with the sir, and sir have many publications in various journals like papers in SCI journals with having average impact factor of 2.85. Total 53 papers are published by sir and 12 book chapters to his credit. He also workshop. He has also completed workshops that are 2 workshops are completed by him and attended 31 conferences. And 45 papers were presented by him in various national and international conferences and 13 invited talks in these conferences. Sir have various life memberships like Marathwada Botanical Society, Maharashtra Society of Genetics and Plant Breeding, Indian Society of Genetic and Plant Breeding, Indian Society of Plant Pathologists, and Indian Ecological Society. The area of interest of research of sir is bioremediation and plant microbes interaction. In bioremediation, he works on plastic waste management, oil degradation, pesticide degradation, and bioextraction of metals. In plant microbe interaction, he working on rhizosphere and air mycology, biocontrol of crop diseases, and epiphytic fungi of crop plants and medicinal plants. So now I request to Ade sir to please start his lecture. Thank you, sir.
Thank you
Sir,
very much. Yes, yes, madam.
excuse me, please, sir. May I now request all the participants that hope all of you will cooperate by just keeping mute your mics and opening your videos. And all of you are requested over here to put your questions in our last session of 10 minutes after every session of the question answer session. So you can raise your hands also. You can ask us to unmute yourself and you can put your questions after each session. And also you can put your questions in chat box too. So may I now request, sir, we are eager to hear Ade sir on our buzzword, that is sustainable development. Sir, please.
Thank you very much. Good afternoon to all. Respected Manorachari sir, who has given a wide roadmap of the future pathological research as well as biofertilizers. Respected principal of the college, head of the department, dear participants. So can I share my slide here?
Sure, sir. Just a minute, we will make you post.
So is it visible?
Yes, sir, you can share. Yes, sir.
Okay.
We are able to see, sir.
Okay. So I have chosen a talk regarding biofertilizers and how biofertilizers are making the disease control. So I think this is appropriate to the title of the conference, and we have to proceed with that. So very first of all, biofertilizers, all of us know, these are the microbes which are of various type. Manorachari sir has told already the PGPR. These are the microbes which can be used as biofertilizer, which also involve the nitrogen fixers and plant growth promoters also. So such type of microbes, if we are going to use for the disease control. It will serve the dual purpose. Here the plant growth promotion is one, and another the protection of the plant. So that is carried out by the biofertilizers apart from say other activities. So such type of biofertilizers we have worked on that, and a particular nitrogen fixing microbe, that is rhizobium, that is especially treated as a case example, and this was used for the disease control of the groundnut root pathogen. So my talk will be a specific one that will be highlighting the research carried out in our laboratory. So very first of all, as you know, the plants are going to interact with other organisms. So such organisms may involve the animals, plant themselves, or the microbes that may be pathogenic or beneficial microbes. So pathogenic microbes may have the attack on the aerial parts as well as underground part, and hence the association of both the kind is possible on the aerial part as well as underground part. So aerial part association, if we try to see, then pathogenic fungi may cause disease and develops the disease, you can say, uh, interaction with the plants, and plant are showing immune response against that diseases, which will be leading to the hypersensitive hypersensitive reaction shown by the plant. Some of the microbes or a pest, you can say. So such pests are beneficial. Pests are causing diseases, just like insect pests that will be causing diseases to the plants, and certain insects are beneficial to the reproduction of the plants, which are helpful for the pollination. So here both the type of say pests, both the type of organisms are available. We can't call them as a pest. Here also certain beneficial microorganisms are there and certain pathogenic microorganisms are there. So here AM fungi or rhizobia, PGPR or trichoderma, all these are coming under the beneficial category, while all the parasitic plants which are grown on the root, just like striga, or the pathogenic bacteria or nematodes or pathogenic fungi, all these will be coming under the pathogenic or harmful microorganism. So association of both of these is there, and in response to that, plant has to behave differently. So plant will try to repel the pathogenic microorganism, while plant will try to attract the beneficial microorganism, and hence positive interrelationship is there with the beneficial microorganism. So these are the interaction, mean possible interaction, which are carried out within the plants and fellow microorganisms. So in terms of why these interactions are possible, what is the base of that? If we try to see, then the living organisms we try to classify as autotrophs and heterotrophs. So autotrophs means all of us know that the food material is synthesized by them. So food material synthesis involves all kind of say nutrients, starting from the carbohydrate, which is very basic nutrient, then followed by it lipids, proteins, vitamins, even hormones. So every construction of the molecule that is done by these autotrophs or photosynthetic mechanism, of course, by the plants. So whenever there is a green pigment, that is chlorophyll, so that is available for making the food or synthesis of food, and hence that particular organism gives them status to the status of the autotrophic organism or self food synthesizing organism. That will involve the bacteria, photosynthetic bacteria, or algae, or rest of the group of the plants which are self-sufficient in synthesis of the food material. Now synthesis of food material means what? they have to connect the carbon dioxide, fix carbon dioxide molecules and connect the carbon elements to each other, and formation of the carbohydrate takes place, means carbon chain generation is there, and this carbon chain is retained by the protein, this is retained with the lipids, this is retained in the heterocyclic compounds of say many steroids, or you can say the hormones or enzymes, all of them are synthesized from this basic carbon chain. So that's why plants are given status of autotrophs. Heterotrophs means the food material is obtained by them from the plants, means which are already synthesized photosynthetically. So these heterotrophs, they have to take the ready-made food material which is made by the plant, and such heterotrophs that will involve the fungi, animal, insects, and even human being. So such type of say heterotrophs, they are there. So it is the story of say interaction. Whenever the food material is available, heterotrophs have to consume that, and the microbes which are feeding on the heterotrophs may be in the live situation or live condition, that is known as parasite, or if the dead condition, if it is there, then microbes are also attacking the dead body, and that will develop the detritus mode. So in this case, the decomposition activity, these are run by the microbes, and these are also categorized as heterotrophs. So autotrophs and heterotrophs, if these are compared, then this basic difference is there. Now, autotrophs, although they are sufficient for the synthesis of the food material, but they have to take the help of heterotroph. So, although the heterotrophs are obtaining their food material from the autotrophs, in response they have to give something to the autotroph, and therefore you find the interaction between autotrophs and heterotrophs. Now, what are the possible role played by the heterotrophs for the plant? Let us understand the microorganisms in the soil. So there are many more bacteria that can serve as plant growth promoting bacteria that will be ultimately generating the hormones, growth hormones, which will be facilitating the growth of the plants, and hence this will be the support provided by the heterotroph. The nitrogen fixing bacteria that will be fixing the atmospheric nitrogen, and that nitrogen is after fixing converted into the soluble forms just like nitrate, and these nitrate are supplied to the plants for the synthesis of say proteins. Then phosphate is fixed, phosphate is solubilized by the uh, microbes or AM fungi, and that will be supplying to the plants. So similar to that, all these nutrients which are required by the plants, there is a big list of the nutrients in the plant. So that will be categorized as macronutrients or micronutrients, and all these nutrients are made available in soluble form by the role of say microbes within the soil. So these particular microbes in the soil, that is having a big role of decomposition or recycling the material, and hence we have the phenomenon biogeochemical cycles in our ecosystem. Similar to that, the animals or insects, they are playing their role for the benefit of the plants. We have seen for the case of pollination, they will be helping. Animals, they are helping plants after consumption. They may be consuming the fruit after of so many plants, and after their consumption a specific treatment to the seeds are given, and these seeds are passed through the elementary canal of the animals, and then in their fecal matter the processed seeds will be generated. So such processed seeds will be taking less time for the germination because of this removal of scarification and recalcitration. So such type of activities are also rendered by the animals, And of course the human being can eat the food and remove the seeds from that. That is one of the thing. Artificial pollination is also possible. We can go for the artificial pollination, and plant is benefited for the reproduction. And similarly, we are taking care of the plants, we are growing the plants, we are favoring a particular species. And of course, as a you can say most intelligent animal on this earth, we are also implementing the conservation of the biodiversity. So such all things are beneficial to the plants, and that's why there is a very good interrelationship between the autotrophs and heterotrophs. And hence, although we are dependent on the food, or bacteria is dependent on the food, in response, it has to provide certain nutrients to the plants. And therefore, although it is appearing that there is a indiscriminate, you can say, exploitation of the plant products by us or by fungi, but at the same time we are having role to support the plants, and hence there is a nice interrelationship with each other. So in view of this, the plants and fungi, they are interacting with each other, and with this interaction, they are getting evolved or already evolved during the course of the time. So here, endophytic fungi, they are showing association with the plants, and they are beneficial association. So plants are benefited as well as fungi are benefited, and in combination with the plants and fungi, certain economically important chemicals are synthesized, and these chemicals are being utilized by us. So such type of endophytic fungi, these are found to be beneficial for the plant. Similar to that, exploitative fungi, these are microbes which will be causing harm to the plants, and this will be the two terminal ends of the association. So here there is association which is parasitic, here there is association which is endophytic. So that will be the mutualistic association, and present day the balancing of both of these, that would be there. And because of this balance, the control over the population, that is already established. And if population is controlled, then and then only the life cycle on this earth or regulation on this earth, that will be smoothly carried out, and earth will be having its capacity to tolerate or to nourish all these organisms with its proper capacity. So that is said to be a carrying capacity. So carrying capacity takes care of the population control, and hence for the sustainable maintenance of Earth's atmosphere or Earth's environment, for the betterment of the organism, then that is automatically maintained because of this phenomenon. So therefore, we called our planet as Mother Earth. So Earth is supporting every one of us. Then this is about the interaction between the plants and fungi, especially. So these kind of say positive interactions are there which are beneficial to the plants, or negative interactions are there that may be causing harm to the plant. So if we make these four quarters, so here all these you can say beneficial fungi or benefits from the plants which are rendered, so that will be incorporated in this particular square. So where both the actions, both the roles are positive. So here facultative obligates are there, and mycorrhizas are there. These are permanently residing with the plant tissues, and in response plants are providing them the food material. So such type of both the positive partners are there, then that is said to be a mutualism, or you can say symbiosis. Here mycoheterotropic associations are there, that is one positive means one is supporting for the food material, of course, by the plant, but the fungi or heterotrophs, they will be causing damages to the plants. So this will be the harmful activity. So one positive, one negative is there. So that is said to be exploitative. Then here the plants are causing harm to the fungi, or fungi are also causing harm to the plants. So here the fungi are providing any material to the plant, but this is going to inhibit. This is to be inhibited by the plant. So this is the example of a resistant plant that will be causing killing of the fungi by secretion of various root exudates, so that fungal pathogens will be repelled and remain away from that plant. And here in fourth quarter, you will find the antagonism of the plant or antagonism of fungi. Both of these are available. Antagonism of the plants, that may be there by the plants due to the allelopathic interaction, and here the antagonism of fungi that will be observed because of the role of say other fungi, so which are antagonistic to that particular fungi. So such type of mix type of, or all the four type of say association that is there. And here both the you can say fungi, means pathogenic fungi or beneficial fungi, these are shown for the comparison purpose. So their mode of infection and spreading throughout the plant tissues, that is one and the same. So here there is a cyst formation, aprosorium generation is there, and here hypanthodium, that is hypopodium, that is developed here in the AM fungi. So after germination, they will be providing the branches. In both the cases, their mode of say movement may be the intercellular. So in both of these cases, this movement is possible, and they develop the hostoria like extensions or projections from the mycelia for the sake of absorbing nutrients from the cell, or that will be absorbing the sap of the cell. And here this will be taking the food material, but in return they have to provide the uh, solubilization of the phosphate, and that will be circulated to the plant tissues. So such type of beneficial as well as harmful mode of fungi that is available, and in this way we are able to designate different type of interaction with the plant material. So in pathological books, this is very common phenomenon, that is disease triangle. So on one side there are hosts, on another side there are pathogens or the insects which are causing the damage to the host, or which are living on this host. And third angle is the environment. So if environment is suitable, then and then only this interaction is possible. Or host, if it is not suitable, then the diseases may not be caused. So for creating the disease, the presence or existence of all these three features, that is very much important. And after understanding this disease triangle, we can go for the management practices for controlling the diseases. So this is the basic of that. If the environment is suitable for the infection, then this will be infecting to the host. At the same time, host should be there in mature condition, that should be full of nutrients, then and then only they will be selecting. Otherwise we know that in meristem culture, no, the meristem culture that will be producing the plants which are virus free. Why these are virus free? Because these are meristematic tissue, and they have to generate the nutrients in due course of time. But immediately after say meristem detachment from the meristem, they are waiting, or they have to take time for the synthesis of the food material, or for the storage of the food material, and that's why they are even the virus free. So even the diseases or pests, they need certain nutritious plants or nutritious host, and for their development, environment is also suitable. So such type of disease triangle, if we try in terms of Flour's gene for gene hypothesis, then we'll observe that of course the favorable environment is taken here as a constant factor. So in this favorable environment, there is an interaction between the genes which are present in the pathogen or resistance genes present in the host. So such type of resistant gene and virulent gene, these are the natural way, or in wild type these genes are available. So that's why in wild condition no plant is found to be affected. But as we go for the domestication practices, the wild genes or resistant genes, they are gradually vanishing, and this species which was otherwise in wild condition resistant, that is now turning to the susceptible one. So if this is turning to the susceptible one, then a virulent can cause the disease even in that. And at the same time you will find the variations among the pathogens. Virulent are their, you can say, natural mode or wild mode, but during the course of time they want to have the enough food material. They have to have the alteration in their genome, and whenever they are altering their genome, then they become virulent. So presently the virus epidemic, corona and all that, they are the result of such type of phenomenon. So virulent strains are making themselves virulent because of the spontaneous mutations and all that. So such type of virulent pathogens, if these are formed, and susceptible host if it is there, then there will be the disease development. So in this case, the environmental factors are considered as constant or favorable for both of these. So because of this gene for gene hypothesis, the characterization or specification has been made, and this specification allows the disease formation wherever the pathogens and the hosts are very specific to each other. Apart from that, there are general pathogens that will be causing common diseases to all kind of hosts. So for example, the wilting of the root or the leaf spot, such type of say pathogens that may be causing diseases to the host, and for controlling them, that kind of say controlling agents are also available, which are very generalized agent. And that kind of say resistance means plants are also showing generalized resistance for the uh, prevention of the entry of the pathogen. Say for example, thick cuticle, that is waxy cuticle. So waxy cuticle don't allow fungi to germinate and piercing the thread into the plant tissue. So this is automatically established by the plants. Then secretion of certain antimicrobial chemical or antimicrobial activities we find in some of the medicinal plants. So such medicinal plants, they will be repelling the microbes from the taking of entry, and generalized antimicrobial compounds are generated, synthesized by them, and that will take care of the varied type of or general range of say microbes. So such type of resistance, that is known as horizontal resistance, while a specific pinpoint resistance, if it is there in the host against a specific pathogen, that is said to be a vertical resistance. So such type of resistance and say virulence, that will go hand in hand, and therefore we find the boom and bust cycle or this disease epidemic. So suppose the host is making resistant for this by say recombination or mutation, then accordingly the pathogen is influencing or making themselves into virulent by alteration in their respective allele, and hence boom of say disease may be caused, and then there will be burst because of this no the pathogen will be controlled, because of the resistant pathogen will be controlled, but at the same time the pathogen will develop again the virulence, and again with that the disease may be caused. So in diseases, boom and bust, these are the alternative phases we find. And this is because of the favorable environmental factor or specificity in the pathogens and host, and of course the very basic principle of the living things try to remain alive. So this is the struggle for existence for both host as well as pathogen. So these are making themselves changes within their genome and get adapted to the changing environment. So dynamics is already created. So coming to the specific crop which is worked out in my laboratory, that is groundnut. So groundnut, all of you know, this is very wonderful crop, and because of the discoveries of Carver in South America, so many products from groundnut were possible to form, and that groundnut, because of this variety of products, that has become very much popular. So, groundnut production, if we try to see, then China is the first number throughout the world, followed by India, and other countries are also doing the production of the groundnut. Then in India, specifically the western and southern India, that will be growing groundnut. These are certain hotspots for the growth of groundnut here in Gujarat, and this is the location of say groundnut research station, which is proposed by the ICAR. So such type of wherever the crops are there, in that area only research stations are established. So such type of groundnut is having especially the tropical climate, and hence it is said to be a tropical crop. So this is coming under the oil seed crop. So these are certain varieties of the groundnut which are grown generally in Maharashtra. So for example, AK12, 24, SV11, Vikram, TG1, Phule Pragati, JL24. So such type of varieties are there, and these varieties are providing more or less quantities of nutrients to us, and that's why we are all fond of this groundnut. Even the layman walking along the road, they can offer the groundnut kernels, roasted kernels, very tasty. And some of us, we can just eat groundnut passively because of the time pass or for passing the time, leisure time we can spend. So because of this particular activity, the groundnut is identified as cashew nut of poor people. So such groundnut is very much nutritive. So far as the nutritive value of the groundnut is concerned, in terms of percentage, this is given, or actual values are also given. 29% energy we get, 12% carbohydrates, 46% proteins, total fat 165. That's why it is coming under oil seed category. Cholesterol 0%, and fibers are 22%, which are most important for keeping our digestive system clean. Apart from that, the folates 60%, niacin, this is vitamin, pantothenic acid, pyridoxine, riboflavin, all these vitamins, they are also present in their appropriate quantity. So such type of uh, nutritious crop is this, and that's why this is to be retained and maintained, and we are maintaining it. But what happens? The protection is needed. So here the sources of say minerals that is also provided by the groundnut. So here the sodium, potassium, calcium, copper, all these minor nutrients which we require, because all these nutrients are required for acting as the cofactors of enzyme. So without them, enzymes will not be working, and hence metabolic activity may be stopped. So to avoid that, we have to have the nutrients. So these nutrients are provided by the food plant, of course, and groundnut is better in this case. So here the again there are certain varieties which are showing particular nutritional status or more or less alteration in their genomes, which will be responsible for different kind of characteristic features. So although this is a situation, groundnut is a major crop and we are fond of groundnut to eat, but this groundnut is subjected or this is affected because of the diseases, certain soil-borne diseases. So root is one of the disease. It is a seed-borne disease. So here many pods, if you try to open, then seeds are said to be affected with the growth of say aspergillus flavus, and because of this aspergillus flavus, apla, this word come, and that's why originated from these aspergillus flavus, the disease is known as apla root disease. So apla root disease, that is rottening the root of groundnut, and because of this rottening process all the absorption is hampered, and hence you find very stunted growth of the groundnut plant over here. So here this is the diseased plant and this is a healthy plant. So by which we can say that the groundnut is affected because of this seed-borne or soil-borne disease, because of this Aspergillus flavus pathogen. Then apart from that, it is very much specific in production of the mycotoxin that is known as aflatoxin. So aflatoxin is deadly poisonous. The story in history of plant pathology, if we read out in the books textbook, it is mentioned that because of the consumption of these infected kernels, the ducklings were died. So that particular reference time to time is given in the pathology books. So such death of these ducklings was because of the consumption of say aflatoxin. So this is a you can say significance of this aspergillus flavus. So if it is there, it will be not only spoiling the kernels, but the growth is also found to be reduced, and hence the crop may be gone, which will be leading to the heavy loss of the groundnut. Then another disease was worked out in our laboratory, that is dry root rot of groundnut. So dry root rot that was that is created by the Aspergillus niger, and that is also known as Aspergillus crown rot or collar rot. So here the roots are dried instead of say turgid condition. The roots are very flaccid and constricted, and because of that, the absorption phenomenon of the groundnut plant is hampered because of this ill development of the root system or damaging, you can say, tissue of these roots. So because of that, the plants will not be supported longer, and immediately after the germination, the plants are going to die. So such groundnut diseases were treated, they were worked out in our laboratory. Then, so far as the control of such diseases, what are the possible methods? So, as Manora Chari sir has explained that there are many more practices which can be incorporated in the form of integrated disease management. So, these are the possible practices for the control of disease or management of disease. So alteration of crop practices, that is very much important. So if there is a specificity of the pathogen and crop, then by altering the crop, means we are altering the habitat of the crop or agricultural land of the crop or sector of the uh, agricultural land, then automatically there is a control over this, because specific host is not available for specific pathogen, and hence the crop will be saved. Then growing disease resistant variety. So disease resistant varieties are available. If there is a particular disease epidemic, then breeders, they used to grow or develop the varieties resistant against that epidemic only. In agricultural system, there are categorization. Most vulnerable disease or very epidemically found disease or minor diseases. So such diseases are categorized, and according to the need of the time, the breeders, they go for the development of different varieties which are specifically resistant to that particular disease. So such type of varieties are available, and if we try to grow those varieties, then automatically there will be disease control because there is a lot of resistance mechanism in this variety. Then next is a chemical control. We have grown the crops, crop is there in the field and it is affected by some of the insects or some of the pathogens. So in that case, we can't afford the uprooting of the crop. So in that case, we have to use the chemicals, and there are many more synthetic chemicals that can be used for the uh, you can say controlling these pathogens or controlling these microbes. So or pest, so several pesticides may be of different category, insecticides, fungicides, or you can say the bactericides or virucides, these are also available. Then coming to the biocontrol, here the designing of specific microorganisms for the control of say pathogenic microorganism. So this is also possible. The originally existing soil competition of the microbes within the soil. So this is to be explored for the biocontrol activity. Originally, already existing mechanism is there regarding the antagonistic microbes that is showing antagonism with each other. So that is also involving the nitrogen fixing microbes also. And if we consider the PGPR, that is plant growth promoting rhizobacteria, then they are also doing this particular job, and especially the species of pseudomonads that will be very much active as say biocontrol agents or antimicrobial agents or antipathogenic agents or antagonistic agents. So such type of microbes one can use for the biocontrol activity, as it is mentioned by Manorachari sir in the morning. Then if you see the ecology of nitrogen fixing bacteria, then we'll see the, just a minute. So such type of nitrogen fixing microorganisms, they are doing the fixation of nitrogen in this form, and because of that the symbiosis is established. So here the rhizobium, if it is there, then they are forming the nodules. Azospirillum, they are the rhizoplane microbes, and here free living nitrogen fixing microbes, they are there. So here the energy is provided by the plants for the multiplication of them, and in return they are providing the nitrates, or at least they support for the nitrogen fixation here. That is addition reduction of this nitrogen and fixing in the form of say ammonia or ammonium ion. So for this particular activity, the nitrogen fixation will be associated with the plants very much as an symbiont, either maybe in the rhizosphere format, or they are free living but they are aggregated with the root system of the crop. Then they can form the association on the plane just like azospirillum, and they are taking entry very integrally, they can form the association with that. So such type of symbiosis or this association or free living aggregated in the rhizosphere zone, such type of nitrogen fixing microbes are there, and they are therefore beneficial for the plants anyway, because of their unique property of say fixing atmospheric nitrogen. So such nitrogen and carbon, these are exchanged because of this association. And now we have to concentrate on rhizobium. This particular bacterium has been tried by our laboratory. So rhizobium, it is very much a popular bacterium for the nitrogen fixing mechanism. So that will be fixing atmospheric nitrogen and convert it in the ammonium ion because of the unique enzyme nitrogenase. And after that, the nitrobacter that will convert this ammonium ion into nitrates, and these nitrates are made available for the plant for the absorption. So such type of rhizobium, this is getting attracted by the plants by the secretion of say deoxy sugars, or certain flavonoids are secreted by the plants, and these are actually attracting the rhizobia to their root system. And after that, the infection is carried out through the root hairs, and the nodules are formed. So such nodule forming nitrogen fixing bacterium rhizobium, that will be associating intimately with the leguminous plants, because there is a specificity, and hence the nodulation is also formed. So nitrogen is fixed by the NEEP genes present in the rhizobium. For the formation of nodules, the nod factors are working, and for the entry of for taking uptake of the hydrogen, which is otherwise toxic to the nitrogenous activity, so that is made by the HUP gene, hydrogen uptake gene. So such genes or assemblies, or you can say operons, they are there in the rhizobium, and that will be working for the better nitrogen fixing phenomenon, and ultimately that will be providing this nitrogen to the plants. So such rhizobium that is associated with this nodule, and if red colored nodules are there, that is showing the more presence of say leg hemoglobin, and that is the indicator of more say population of the rhizobium. So such rhizobium that can be isolated, that can be maintained, and that will be used as a biofertilizer. So this is the typical example of say biofertilizer um, established or prepared, manufactured from the rhizobium say bacterium. So for the making of biofertilizer, the process is very easier. Just isolate the rhizobium on yeast extract, mannitol, agar medium, or any nitrogen free medium. If you supply, the nitrogen fixers can be multiplied on that, and after that the inoculum is developed, inoculum is multiplied, and that is mixed with the lignite soil, and after that such type of packing will be carried out, and this is sold in the market as biofertilizer. So same biofertilizers can also be acted as I can say biocontrol agents. So how, let us see further. So this is a routine process of say isolation of rhizobium from the root nodules on the sterilized medium. Then here this will be inoculated on the plates, and such type of colonies that will be obtained. So red color is because of the Congo red, which will differentiate the agrobacterium from the rhizobium, and in this way we can have the pure culture of rhizobium. So such pure culture we can use for mixing it with the lignite soil, and which is acting as carrier, and that is available in the form of say biofertilizers. So such type of rhizobium that is available in almost all the legumes, even the crops as well as wild plants. So in our laboratory, we try to use the rhizobia from the wild plant, and these were the wild plant which we have selected: Indigofera linifolia, Indigofera trifoliata variety Duthi, then Alicicarpus tetragonolobus, Tephrosia pumila, and for controlling the groundnut pathogens, it was compulsory to obtain the rhizobium from the groundnut also. So such type of rhizobia can be classified into different categories based on the infecting you can say host. So if infection to the done to the host glycine mac, then that is known as rhizobium japonicum. If infection is there on the trifolium, alexandrium, rhizobium trifoli, If infection is done on the P, Physum sativum, then Rhizobium leguminosarum, then Melilotus alpha, alpha alpha. If it is the host for the nodulation, then Rhizobium meliloti. Then Arachis hypogea is the groundnut. The Rhizobium is grouped as miscellaneous. Then lupinus, this particular species, if it is infected by the rhizobium formation of the nodule, then Rhizobium lupini is the species. So such type of rhizobium and legume association is there, and even in that the specificity is there. So that is said to be the host specific rhizobia. So such host specific rhizobia were tried to uh, experiment, and the nodulation was observed on the groundnut plant. So this, fortunately, all these were found to be nodulating the groundnut plants. It means that they belong to the miscellaneous group, because groundnut rhizobia, they were found to be belonging to the miscellaneous group. And then the other, you can say, characteristic features of the groundnut plants that was observed in terms of, say, number of nodules on main root, side root, or total nodules on the entire root. Then dry matter per plant was calculated, nitrogen fixed per plant that was calculated, and this is the percentage nitrogen. So if we try to supply anything, means nitrogen fixer, then it is our duty to take the care of this, and performance of this is to be assessed. And here we have done this particular activity, and nitrogen fixation or nitrogen percentage we could calculate. Then Pseudomonas fluorescens is another bacterium present in the soil that is having the control properties, biocontrol properties over the pathogenic microbe. So it was targeted towards the control of, say, pathogens, apleroot, that is Aspergillus flavus, and dry root rot pathogen Aspergillus niger. So these fluorescent Pseudomonas that was applied on that, this is already known for the biocontrol agent or biocontrol property. So this was isolated on the King's medium, all these you can say biochemical tests, as well as this particular specific test of obtaining the fluorescence in a UV light, that was also done, and such was tried on the pathogens. So these are the photographs of, say, Aspergillus flavus, and these are the Pseudomonas bacteria. So very good antagonistic zone that was done, that was observed. Here the Aspergillus flavus against the Niger, that was also observed as antagonistic zone. So here the antagonistic zones are given, that is zone of inhibition in terms of, say, minimum millimeter distance or that range. So here Pseudomonas fluorescens 1, 2, 3, these three isolates were treated with the Aspergillus flavus and Aspergillus niger. So this is showing similar activity, similar zone of inhibition. So by taking this particular property of, say, Pseudomonas fluorescens into consideration, we try to introduce that particular activity to the rhizobium, so that rhizobium will become the biocontrol agent. Uh, the Before the combination of Pseudomonas and biocontrol rhizobium, the biocontrol activity was not up to the mark, but that's why we have decided to transfer the genetic material from the Pseudomonas to the rhizobium, uh, and hence this particular mechanism of, say, parasexual mode of bacterial life cycle that was used. So parasexual mode of bacterial life cycle make use of the transfer of the gene or transfer of the DNA from one bacterium to another. So the wherever the donor bacterium is there, from that the transfer is made, and these are the recipient bacteria which receive the DNA from the donor bacterial cell. So there are three possible mechanisms for the reception. If naked DNA is released from the donor cell and if it is taken up by the recipient cell, this is said to be the transformation. If this DNA is encapsulated here with the genome of virus, and this virus or bacteriophage, if that is taking infection, that is carrying infection and injecting this DNA to the recipient cell, and that DNA is going to integrate with the genome of this recipient cell, that is said to be a transduction cycle. And conjugation is the phenomenon wherever there is a formation of conjugation tube, and here from the donor cell certain genes are transferred to the recipient cell. So why such bacteria they can have such type of mechanism which is known as parasexual mode of life cycle? Within the same generation, if one bacterium is facing problem, then that problem is resolved by the fellow bacterium. These fellow bacteria are known as donor cells, while the problem facing bacteria are known as the recipient cell. So just like human being, no, we try to help each other within the same generation. We are colleagues and we are helping each other by donating some required things by them. So within the same generation, it is to be carried out, and we also know that we are getting certain benefits from our parents. So just like that, the bacteria, they are also getting certain benefits from their respective parents. So by binary fission, they are multiplying, and they become they continue that kind of race further. But at the same time, they also exchange their material if this is needed by a needy bacterial cell. So because of this particular, you can say, mode of transfer of genes, that is said to be parasexual mode of bacterial life cycle. So we have tried to use this parasexual mode of bacterial life cycle, and out of these three, the conjugation phenomenon was selected by us. So here, the conjugation phenomenon that was selected because we can do it easily. Uh, the antibiotic resistance that is borne by the plasmid, which is present here in the bacteria, so such plasmids that may be there here in the donor bacteria, that will be transferred to the recipient bacteria, and recipient bacteria don't have such type of plasmid over there. So if we keep this bacterium, donor bacterium and recipient bacterium side by side, then they establish the conjugation tube, and from this donor bacterium, the copy of the plasmid is getting transferred to the recipient bacterium, and the recipient bacterium is now the resistant against that particular antibiotic. So such type of conjugation tube would be formed here. There will be enhanced, you can say, transfer of the plasmid, and after transfer of the plasmid, both the bacteria are resistant against that antibiotic. So this is a routine phenomenon of the bacterium. Such phenomenon we have explored, and that is termed as transconjugation because the bacteria were different. So here donor bacterium was the Pseudomonas fluorescens, and receiving recipient bacterium was the rhizobium. So such type of transfer of say resistant gene that was carried out to the rhizobium, and hence that particular term transconjugation is established. So for the basis of bacterial conjugation, the antibiotic sensitivity was used as marker, and such marker if we use, then we have to test the antibiotic sensitivity of each of these. So these were tested, rhizobia and Pseudomonas fluorescens, these were tested for the antibiotic sensitivity, and opposite results we have to finalize. So RTP, that is the rhizobium obtained from Tephrosia pumila, isolate number 1, this was found to be streptomycin resistant, but at the same time, the susceptibility is there for the tetracycline antibiotic. While There was opposite phenomenon in case of Pseudomonas fluorescens isolate 1. This was found resistant to tetracycline but susceptible to streptomycin. So here, if this is a phenomenon and if we have the antibiotic or mixture of both of these simultaneously in a medium, then this problem of survival can be solved only because of the exchange of these genes or exchange of this plasmid. So such type of conjugation was established by them in a LB broth medium. This conjugation was carried out with the different donor to recipient concentration, and then the conjugants were multiplied afterwards with the suitable medium without adding any antibiotic. So this was the plasmid transfer intensity and formation of conjugants that was there. So such conjugants were multiplied. These are known as trans conjugants were multiplied, and these were tested for the antagonism both by the on the plate or as well as in dual culture technique, as well as these were deposited into the root system where these are already infected with the apfla root as well as dry root rot of the groundnut by addition of say Aspergillus flavus and Aspergillus niger, and then we could calculate the percentage control efficacy. So here the variable percentage of say controlling that was shown by the trans conjugants along with the Aspergillus flavus or Aspergillus niger, and the percentage control efficacy is seen like this. So this is a small, this was a small effort to make an establishment of a nitrogen fixer as biocontrol agents. So already this is one of the example rhizobium which we have worked out. So just like that, there is a possibility to have the nitrogen fixers which have the also capacity to control the pathogenic fungi or pathogenic bacteria. So such type of controlling mechanism may be present, and it is the need nowadays to explore their properties for the antagonism. And if it is done, then pesticide free you can say environment can be created, because most of the pesticides are recalcitrant and they are not you can say consumable consumed by the bacteria, or they are not degraded at all. So such type of recalcitrant pesticides, if these are there, then they will be creating the accumulation beyond the level, and if accumulation is there continuously, then that will be circulated to the plants, and we are dependent on the plants, therefore that will be circulated to us, and we have to face many more problems due to this pesticide. So such type of bigger problem that can be controlled at the smaller level with the usage of biocontrol mechanism, and hence we this particular mechanism will be one of the uh, field for the sustainable agriculture, because we are very much careful, we should be careful about our future generations. So our future generations, if they are expecting from us a contamination free soil, then we can maintain for them, we can keep from them, and this may be helped by the nitrogen fixer, and rhizobia has already provided the help for this. So with the help of these microbes, we can maintain our sustainable agriculture, and the sustainable development, which is a part with agriculture. Sustainable agriculture is a part of sustainable development. So for longer time, we can sustain our population, and we can maintain our race on this earth. So with this, I'm very much thankful to the organizer for giving me an opportunity to deliver lecture in this particular seminar. Thank you very much.
Thank you so much. Thank you so much, sir, for your esteemed guidance. Now, question answer session. Any question from audience, please? Those who have any question can unmute yourself and ask the question. I think no question is there. Thank you so much, sir, for providing many important insights to all of us. Extremely knowledgeable session really is all for all of us, sir. Starting from food chain, from autotrophs to heterotrophs, in interaction and their interrelationship between them through plants, you have directly come, you have taken us, and through the travel, you have arrived us to how the rhizobium can effectively be used as a biofertilizer as well as a biocontrolling agent. Thank you so much, sir, for your important insights. You have made us aware about the use of the biofertilizers and biopesticides for sustainable development. Thank you. On behalf of all the trustees, Amulak Jain Vidya Prasarak Mandal, as well as on behalf of our dynamic principal, Dr. N.S. Rathi sir, I take this opportunity to extend my deep sense of gratitude towards you, sir. Thank you. Thank you, sir.
Thank you so much, madam.
Now, hereafter, I would like to, it is my pride privilege once again to welcome over here and to invite Professor Dr. Umesh Mogle from Jalna to enlighten us. Sir, please.
I am audible. I am audible.
Yes, sir, you are audible. Please, technical help I need. Please give spotlight to sir.
Okay.
Yes. May I now request Sayyed sir to come forward to introduce Umesh Mogle sir. Okay, Patale sir, I would like to request Patale sir to come forward to introduce Umesh Mogle sir.
Good afternoon. It is great pleasure to introduce my friend, Dr. Umesh P. Mogle, Head Department of Botany.
Sir, you are not audible. Patale sir.
Qualification is MSc, PhD in botany. Teaching experience for Professor Umesh P. Mogle, 18 years, and research publications, 46 research papers in reputed national and international journals. The research area, antagonistic effect of fungi and bacteria, biological control, biofertilizer, soil health and microflora, and weed utilization and management. His research guide, 6 students working under his guidance, and 1 is awarded PhD degree. He is also received 2 major research projects and completed funded by UGC, New Delhi. Thank you. Thank you, sir.
I would like to invite Mogle sir
to come forward to deliver his lecture. Sir, please.
Okay, I am audible.
Yes, sir, you are audible.
Okay,
Yes,
okay.
sir.
Okay, let us start.
Sir, your voice is very low. Would you be able to use, sir, headphones?
Headphones, ear headphones. Now,
Yes, sir. Still a little bit disturbances are there.
okay.
Means your voice is very low.
I think there is a problem in what I know.
Okay,
low
sir,
voice
you
is
can
there?
continue.
Okay, let us start. One day conference organized by Department of Botany. So that is the nice activity. So today's inaugurated respected scientist, Professor C. Manorachari. Today's speaker, Professor Avinash Ade sir, Head Department of Botany, Savitribai Phule University, Pune.
Sir, just a minute.
Next speaker is Dr. B.D. Takate sir, Nevasa, Organizing Committee. So respected Principal Professor Dr. N.S. Rati sir, Dr. J.M. Bhandari sir, Vice Principal, Mr. I.G. Sayed sir, Head and Convener of the National Conference, Dr. P.S. Anarse sir, Co-Convener of the National Conference, Dr. S.S. Patay sir, Organizing Secretary of the National Conference, all dear participants, friends and colleagues. So now it is the COVID-19. So due to the COVID-19, so situation is very bad. So due to the COVID-19 situation and in view of the safety view of the student, participant, researchers, so there is this year all the activities will be conducted in online mode. So one of the part of that, so our Department of Botany organized a nice conference online. I heartily congratulate the organizing team. So national conference on role of, so my topic is on role of biofertilizer for sustainable agriculture. On that, my specifically I am speak on the topic that is biofertilizer, a modern approach for organic farming. I wish to share my presentation.
नाही माझ्या रूमवर जा ना आणि तिथून त्यांना म्हणा हेडफोन सांगितला म्हणा अक्षयला लवकर ये जाऊ.
Respected Mogle sir, just would like to convey your message that you are allotted with 30 minutes, sir, please.
Okay, okay. I will finish in 30 minutes. Thank you.
Thank you, sir.
My topic is biofertilizer, a modern approach for organic farming. So content of presentation starts the here introduction of organic farming. Within that, biofertilizer, types of biofertilizer, application of biofertilizer, biofertilizer precautions. In B section, I would like to present geographic producer and distribution, that is in growth, productivity, long-term studies, profitability of biofertilizer, labor and employment of the biofertilizer, Millennium Development Goal, and overview research of in our India. And last slide, there is the conclusion and references. So let us start from the few words of Papamal Rangamal. So I don't know anything about agriculture. So these are the few words of Papamal. I don't know I know anything about the agriculture. I have never had a formal education, but I have still come so ahead in life, seeing my work and dedication. They have given this award. So that is the only one Padma Shri awardee, that is Pappamal Rangmal, speaking to the media on the occasion of being awarded the Padma Shri Award on the 72nd Republic Day of our India. So this is the uh, Padma Shri awardee, Pappamal. So age of the Pappamal is here 105. So that is from the native from the Coimbatore, who runs her own organic farm, confirmed Padma Shri Award in 2021. So these are the beautiful pics of awardee. That is the Padma Shri awardee, Papamal with PM. So here is the her farm of Papamal. So, and that is the field of banana. So who is the Papamal? Just I introduce, because her great work on organic farming. So Papamal, born 1914, is an organic farmer from Tamil Nadu in India. At the age of 105, she is argued to the oldest farmer. So she is argued as the oldest farmer still active in the field. She regarded as a pioneer in the agriculture field and is affiliated with the Tamil Nadu Agricultural University Department of Education. At her age, she works every day on her 2.5 acres land. So that is the great discovery of the Pappamal. So the government of India honored her in 2021 with the fourth highest civilian award of Padma Shri for her role in organic farming. Okay, so few words of the Pappamal. That is, I eat organic ragi. So Pappamal says, so I eat organic ragi and millets and have never taken any medicine yet. So because that is organic food. So that is the benefit of organic food. So that Pappamal has at any medicine. I have never fallen sick and I am always healthy. Okay. So this is the importance of agriculture, organic farming. So that is, Pappamal is healthy at their age, that is 105. Since 1970, I have been eating food of banana leaves only. Okay. So, and that is medicine free. This is very healthy and even helps our hair grow longer and stronger. So these are the few words of the Pappamal. So now question is arising now that what is meant by organic farming. So this is very basic question. That is, what is meant by organic farming. So the Ade sir just nicely introduced our topic, that is biofertilizer. So that is the one of the part of that. So what is meant by organic farming? So cultivating the land and raising crops in such a way that the soil is kept alive and in good health by use of organic waste. Okay. So here important word is alive. So soil, that is soil is kept alive, means nowadays using of the chemical fertilizer, so that is the overdose on the soil, and that soil is unfertile, so that is soil is dead nowadays. So every citizen has their role to keep their soil alive using the organic waste in their field. So here crop, animal and farm waste, aquatic waste and other biological materials. So that is used in their field. So Franklin D. Roosevelt's words of the Franklin, a nation that destroy its soil, destroy itself. So these are the words of Franklin. So this is the very good message for all the uh, world given by the Franklin. So Jaggi Vasudev. So another quote of the Jaggi Vasudev is very nice. So with wrong farming methods, we turn fertile land into desert. Unless we go back to organic farming and save the soil, there is no future. If we not using the organic farming, so that time there is no future of that soil. Okay, so this is the very good quote of the Jaggi Vasudev. Okay, just what is meant by organic farming? Organic farming is defined by the use of fertilizer of organic origin such as compost, manure, green manure, and bone meal, and places emphasis on techniques such as crop rotation is very needed. So rotate the crop, that is one field to the another field, that is also important. So that is leguminous crops and cereals crop alternatively rotate, and that rotating and with helping of the crop rotation definitely increase the fertility of the soil. And next, organic farming advocates claim advantages in sustainability, openness, self-sufficiency, autonomy, independence, health, food security, and food safety. Okay, so here these are the advantages of organic farming. So that is self-sufficiency, autonomy, in independence, good health. So that is the food security and food safety. In detail, view of the organic agriculture can be defined as an integrated farming system that strives for sustainability, the enhancement of soil fertility, and biological diversity. Okay, no doubt, I think that is the organic farming. So when we applying the compost in our field, so that means increasing the diversity of that soil. Okay, so while with rare exceptions prohibiting synthetic pesticides, antibiotics, and synthetic fertilizers. Okay, so genetically modified organisms and growth hormones also. Since 1990, the market for organic food and other products has grown rapidly. So reaching 6,300 crore worldwide in 2012. Okay, nowadays, so there will be the awareness in people. So why we are using the chemical fertilizer in their field. So importance of organic food is nowadays increase. So that just they are demanding the organic food. Okay, so this demand has been driven a similar increase in organically managed farmland that grew from 2001 to 11 at compounding rate of 8.9% per annum. Okay, so just they increasing the percentage per annum. As of 2019, approximately, that is 7.23 crores hectares 7.23 crore hectares worldwide were farmed organically. So representing the approximately 1.5% of world farmland. So just that increasing the cultivation area of the organic farming due to the benefits of the organic. So organic farming relies more heavily on the natural breakdown of organic matter than the average conventional farm, using techniques like green manure and composting to replace nutrients taken from the soil by previous crops. Okay, so that is long-term effects also from that organic farming. We get the result that is in their long-term effect. So this biological process driven by the biofertilizer and microorganisms such as mycorrhiza and earthworms, that is the vermicompost, release nutrients available to plants throughout the growing season. So we move toward the biofertilizer. So biofertilizers are natural fertilizers that are microbial inoculants of bacteria, algae, and fungi. So in combination form of that bacteria combined with algae, and that bacteria also combined with fungi. So that is the we are applying the, that is the in combination form also, which may help biological nitrogen fixation for the benefit of the plants. They help build up the soil microflora. So build up the microflora as well as, that is microflora increase due to the that combination of that bacteria, algae, and fungi, and applying the organic manure. So definitely it increase the soil microflora and thereby the soil health. So biofertilizers also include organic fertilizers. Use of biofertilizer is recommended for improving the soil fertility in organic farming. Okay, so that biofertilizer are not showing alone their effects. If we have to combine use that biofertilizer and organic, that is organic manure, so definitely increase the productivity. So a biofertilizer is a substance which contains living microorganisms when applied to the seed. So when externally we have to apply the biofertilizer to the seed, so plant surfaces or soil colonizes the rhizosphere or the interior of the plant and promotes growth by increasing the supply or availability of primary nutrients to the host plant. So that's important. So biofertilizer add nutrients through the natural process of nitrogen fixation, solubilizing phosphorus and stimulating plant growth through the synthesis of growth promoting substances. So too many types of biofertilizer. So bacterial, fungal, algal, aquatic fern, earthworms, that is vermicompost, and VAM fungi. So particularly we have to classify that biofertilizer. So that is bacteria is one of the biofertilizer. That is symbiotic nitrogen fixers are there. So popular examples are Rhizobium, Azospirillum. So Azospirillum, too many species are there. So that is symbiotic nitrogen fixers are there. Along with that, there will be the free living nitrogen fixers are also there. So that is Azotobacter, Klebsiella. So these are the few examples of free living nitrogen fixers. So on view of the above, so algal biofertilizers are also important. So that is popular examples are blue-green algae. So in association with azolla, so that is very important. In association with azolla, anabina, nostoc, oscillatoria, these are the few examples of algal biofertilizers. So that is incorporated with the field, particularly that is paddy field, that is blue-green algae. So the next one is phosphate solubilizing bacteria are also there. So popular examples are Pseudomonas, Bacillus. So fungal biofertilizer are there. That is popular example is FAM, and that is earthworm producing the, that is also that is vermicompost. So when we view of the above, so bacterial biofertilizers are also important. The live cell of bacteria used as biofertilizers. These microbes contain unique gene called as NIB gene, which make them capable of fixing nitrogen. So that is NIB gene is responsible to fixing the nitrogen. The nitrogen fixing bacteria work under two conditions. So there will be the two conditions will be there, that is symbiotic condition. So that is free living bacteria. Non-symbiotic, the symbiotic bacteria make an association with the crop plants through forming nodules in their roots. That is very popular. That is leguminous plants are producing the root nodules, and they convert the free atmospheric nitrogen into nitrites. The free living bacteria do not form any association, but live freely and fix atmospheric nitrogen. So that is important. Then symbiotic nitrogen fixers are there. So most important symbiotic nitrogen fixing bacteria is Rhizobium and Azospirillum. So that is symbiotically associated with the plant and fixes the nitrogen. So that is very popular example is Rhizobium. So Rhizobium lives in the root hairs of the legume. So that legumes, that is Rhizobium lives in the root hairs of the legumes by forming the nodules and convert the atmospheric nitrogen. So here plant roots supply essential minerals and newly synthesized substances to the bacteria. The name Rhizobium was established by Frank in 1889. This genus has 7 distinct species based on cross inoculation, that is more than 20 cross inoculations groups have been established. That is a new classification has been established for Rhizobium. This is slow growing Rhizobia known as brandy Rhizobium, and other group is fast growing Rhizobia called Rhizobium. So these are the different peaks, that is Rhizobium bacteria inside the nodules. So this is the Rhizobium inoculated in the laboratory in a nutrient plate, and these are the root nodules of leguminous plants. So Rhizobium can fix 50 to 300 kg per hectare, applying in 50 to 300 kg per hectare. Then very popular biofertilizer is Azospirillum. It is mainly present in cereal plants. It is mainly present in cereal plants. So inhabits both root cells as well as surrounding of roots. So azospirillum forming symbiotic relation and increasing nitrogen fixing potential of the cereal plants. So azospirillum is recognized as a dominant soil microbe. So this is dominantly present in the soil. So around the that is cereals. So nitrogen in the range of 20 to 40 kg per hectare in the rhizosphere in non-leguminous plants such as cereals, millets, oil seeds, cotton. So these are the crops and their surrounding, that is the azospirillum is present. So here considerable quality of the nitrogen fertilizer up to 25 to 30% can be saved by the use of azospirillum inoculant. So these species have been commercially exploited for the use of nitrogen supplying biofertilizers. The benefit of plants by inoculation with azospirillum have been primarily attributed to its capacity. So that is fixed atmospheric nitrogen, but also to its capacity to synthesize phytohormones also. So this is the additionally synthesize the phytohormones, in particular indole 3 acetic acid. That is azospirillum produces the indole 3 acetic acid. So around the soil of cereals. Recently, an increasing number of studies has attributed an important role of azospirillum in conferring to plant tolerance of abiotic and biotic stresses, which may be mediated by phytohormones acting as a signaling molecules. So particular mechanisms will be there. So mechanisms of tolerance of biotic and abiotic stresses induced by the azospirillum in plants. Okay. So biotic and abiotic stresses will be there. So tolerance to the biotic stress include induced systematic resistance mediated by increased levels of phytohormones in the jasmonic acid, ethylene, so pathway independent of salicylic acid and systemic acquired resistance. A mechanism previously studied with phytopathogens controlled by the intermediate levels of acid. So tolerance of also it is biotic as well as abiotic also there. So tolerance of abiotic stresses will be there named as induced systemic tolerance is mediated by the antioxidant osmotic adjustment, production of phytohormones and defense strategies, what it may be. So such as the expressions of the pathogenesis related to the PR genes. So this is the actual diagrammatic representation of the mechanism of tolerance. So here synthesis of pre-protein and phytoalexin, that is protection against phytopathogen. So here these are the root growths and water stresses, that is salinity stress will be there. So that is increase potassium uptake. So that is antioxidant enzyme in abiotic and biotic stress. So this is the overall mechanism of tolerance. So tolerance of biotic stresses is controlled by the mechanism of an induced systemic resistance mediated by the increased level of phytohormones in the jasmonic acid ethylene pathway independent of salicylic acid. Whereas in the systemic acquired resistance, a mechanism previously studied, we have two, that is phytopathogens also inhibited by that. It is controlled by the intermediate level of acid. So azospirillum can also promote plant growth by mechanisms of tolerance of abiotic stresses named as induced systematic tolerance mediated by antioxidants. So osmotic adjustment, production of phytohormones and defense strategies such as the expressions of pathogenesis related gene. So this is the table showing that is the behavioral type, that is phytostimulant, that is nutrient mobilizer, biofertilizer and biopesticides, and their functions. So these are the popular examples, and these are the references of the who used the phytostimulants and nutrient, biofertilizer and biopesticides. So next, biofertilizer is azotobacter. So free living bacteria, that is large number of free living or non-symbiotic bacteria present in soil. Commonly used free living bacteria are Azotobacter clavicula. It will be not associated with the plant, so that is freely present within the soil. So Azotobacter is a biofertilizer which provides the required amount of nitrogen to the plant from the soil. So many strains of Azotobacter also exhibit fungicidal properties against certain species of the fungus. So response of Azotobacter has been seen in rice, maize, cotton, sugarcane, pear millet, vegetable and some plantation crops. So it improves seed germination and plant growth. Azotobacter is heaviest breathing organism and requires a large amount of organic carbon for its growth. So mass production of Azotobacter, preparation of inoculation packets. So these are the preparation of inoculation pockets, preparation of pockets. Next one is vesicular abrascular mycorrhizae. So all of you know the vesicular abrascular mycorrhizae. The term mycorrhizae was taken from the Greek language meaning fungus root. The term coined by Frank in 1885. So already we have that is a lecture that is Professor Dr. Manorachari taken this topic very detailed. So I'm skip this RAM. So algae as a biofertilizer. So another group of free living nitrogen fixers are cyanobacteria, commonly called as blue-green algae. More than 100 species of blue-green algae can fix nitrogen. So nitrogen fixation takes place in specialized cells called heterocysts. So that is blue-green algae, very common in rice field. So with the help of that heterocyst cells, so that fragment of the nostoc also divide, and there is the reproduction, that is the rapid production of the that is blue-green algae from the heterocyst cell. So unlike Azotobacter, that is blue-green algae are not inhibited by the presence of chemical fertilizers. So no chemical fertilizer added. Inoculation of the algae can result 10 to 14% increase in crop field. So algae, so these are the different algae easy to produce. So usually they are mass produced in cement tanks filled with fresh water. So that is the availability of algae easily in our surrounding. So not requires any processing, quiet and cheap method. So growing of the algae cost of 10 kg maybe rupees 30 to 40 only. Okay. So that is sometimes algae are called as the weed. Okay, and weeds are always troublesome. So beneficial in certain, but it is beneficial in certain crops like vegetables, cotton, and sugarcane. So that is some algal biofertilizers are Anabina, Nostoc, Oscillatoria. So that is next one is Azolla as a biofertilizers. Okay, so this is very popular algae, and that is that Azolla is a tiny freshwater fern. Sorry, common in ponds, ditches, and rice fields. It has been used as biofertilizer for rice in all major rice growing countries, including India, Thailand, Korea, Philippines, Brazil, and West Africa. So that is particularly I'm speaking on that Azolla biomass gets doubled within 5 to 7 days by vegetative methods. Fix 40 to 80 kg nitrogen, that is per hectare per year. So good manure for flooded rice. So increase of crop yield when we applying the Azolla up to 15 to 20% has been observed while fertilizing the rice with Azolla. So tolerant to the heat and cold, fixed, that is 4 to 5% more nitrogen. So biofertilizer applications methods, there are 3 ways of using these nitrogen fixing: bacteria, seed treatment, root dipping, soil applications. So seed treatment, particularly in seed treatment, is the most common method adopted for all types of inoculants when we externally applied the seed treatment to the seeds. The seed treatment is effective and economic. Seed treatment with Rhizobium, Azotobacter, and Azospirillum. So biofertilizer precautions. Store biofertilizer packets in cool and dry places away from the direct sunlight and heat. So maintain the that is humidity. Use the right combination of biofertilizer. So Rhizobium is a crop specific, so use in specified crop only. So that is why it is specific, because leguminous crop, so only that is the Rhizobium applied on the leguminous crop. Do not mix with chemical. Use the packets before expiry only on the specified crop by the recommended methods. So advantages of biofertilizer, that is renewable sources of nutrients, sustain soil health, supplement chemical fertilizers, replace 25 to 30% chemical fertilizers, increase the grain yield by 10 to 40%, decompose plant residues and stabilize CN ratio of soil, improve texture, that is structure and water holding capacity of the soil, also no adverse effect on plant growth and soil fertility. So some of the disadvantages of the biofertilizers are there, that is biofertilizers require a special care for long-term storage because they are alive, must be used before their expiry date. If other microorganisms contaminate the carrier medium, or if growers use the wrong strains, they are not as effective. Biofertilizer lose their effectiveness if the soil is too hot or dry. Next, so that is geographic producer distribution. The markets for organic producers are strongest in North America. So I will discuss here worldwide of the geographic producer and distribution. The markets for organic products are strongest in North America and Europe, which as 2001 are estimated to have $6 and $8 billion respectively of the $20 billion global market. We have the Asia has 9.5%, while North America has 7.2%. Africa has 3% only. So besides Australia, the Australia countries with the most organic farmlands are Argentina, then China, and United States. Much of Argentina, that is organic farmland, is pastures like that of Australia. So here Spain, Germany, Brazil, that is Uruguay and England follow the United States in the amount of organic land. In the European Union, that is 3.9% of the total utilized agriculture area was used for organic production in 2005. The countries with highest proportion of organic land were Austria, that is 11%, and Italy, that is 8.4%, followed by the Czech Republic and Greece, that is 7.2%. The lowest figures were shown for Malta, that is 0.2% only, and Poland, that is 0.6%, and Ireland, that is 0.8%. In 2019, the proportion of organic land in the EU growers, that is to 4.7%. That is the countries with the highest share of agricultural land were, that is Liechtenstein, that is 26.9%, Austria, that is 18.5%, and Sweden, that is 12.6%. 16% of all farmers in Austria produce organically in 2010. By the same year, the proportion of organic land increased to 20%. In 2005, 1.68 lakh hectare of land in Poland was under organic management. In 2012, 2.88 lakh hectares were under organic production, and there were about 15.5 thousand organic farmers. Retail sales of organic products were 18 million euro in 2011. As of 2012, organic exports were part of the government's economic development strategy. So these are the some figures of the growth of biofertilizer in different countries. I'll skip this point. So here, this is the figure of showing the growth of organic farmlands from 2000. So these are the African countries. So African countries, that is organic farmland since 2000. Okay, so that is the very lowest. The next European countries, that is these are the European countries. The next North America. So here, this is the North America. Asia is the lowest one, that is at the bottom. Then Latin America. So Latin America and Oceania. So productivity of the organic farming. So studies comprising yields have had mixed results. So that is the difference among the findings can often be attributed to the variations between study design, including differences in the crop studies and the methodology by which results were gathered. A 2012 meta-analysis found that productivity is typically lower for organic farming than the conventional farming, but that the size of the difference depends on context and in some cases may be very small. While organic yields can be lower than the conventional yields. Another meta-analysis published in Sustainable Agriculture Research in 2015 concluded that certain organic on-farm practices could help narrow this gap. Timely weed management is also very needed when we are applying the biofertilizer. That time when we are not manage the weed, so that time productivity of the crop also decreased. And application of manure in conjunction with the legume, forage, cover crops were shown to have positive results in increasing organic corn and soybean productivity also. So no doubt organic farming has a long-term effects. A study published in 2005 so compared conventional cropping. So that is organic animal-based cropping and organic legume-based cropping on a test farm at the Rodale Institute over 22 years. So that is long-term effect of the organic manure, because the organic manures are slowly decomposed within the soil and they're showing their effect. So the study found that the crop yield for corn and soybean were similar in the organic animal, organic legume and conventional farming system. It also found that significantly less fossil energy was expended to produce corn in the Rodale Institute. Okay. So there was a little difference in energy input between the different treatments for producing soybean. So there will be the profitabilities. This point is very important. When we have the when we sowing some crops in our field, that time farmer not getting the profit in that crop. So when using the such types of organic uh, manures in their crops, definitely there will be the profit in that crop. So in the United States, organic farming has been shown to be 2.7 to 3.8 times more profitable for the farmers than conventional farming when prevailing the price premiums are taken into account. So globally, organic farming is between 22 and 35% more profitable for farmers than conventional methods, according to the 2015 meta-analysis of study conducted across 5 continents. So the profitability of organic agriculture can attribute it to be number of factors. Okay, so number of factors will be there. Which factors are there? So weed management is also important when we are applying the organic manure within our field. So that time when weed management will be not there, that time that weeds also uptake the nutrient content which is present within that soil and lowest the productivity. So first, organic farmers do not rely on synthetic fertilizers and pesticide inputs, which can be costly. In addition, organic foods currently enjoy a price premium over conventionally produced foods, meaning that organic farmers can often get more for their yield. So labor and employment. So this topic is very important. So organic farming, so that is organic production is more labor intensive than conventional production. On the one hand, this increased labor cost is one factor that makes organic food more expensive. So that is organic food feed also expensive. So organic, what it may be, the that is the expensive. On the other hand, the increased need for labor may be seen as an employment dividend of organic farming, providing more jobs per unit area than the conventional systems. So the next, World Food Security. In 2007, the United Nations Foods and Agriculture Organization said that the organic agriculture often leads to higher prices and hence a better income for farmers, so it should be promoted. Other analysis by many agribusiness executives, agricultural and environmental scientists, and the international agriculture experts revealed the opinion that organic farming would not only increase the world's food supply but might be the only way to eradicate hunger. So capacity building in developing countries, organic agriculture can contribute to ecological sustainability, especially in poorer countries. The application of organic principles enables employment of local resources, for example, local seed varieties, manure, and therefore cost is effectiveness. Local and international markets for organic products show tremendous growth prospects and often offer creative producers and exporters excellent opportunity to improve their income and living conditions. So Millennium Development Goal, a series of case studies conducted in selected areas in Asia countries by the Asian Development Bank Institute, ADBI, published as a book on poverty elevations by way of higher incomes, improved farmers' health owing to less chemical exposures, integration of sustainable principles into rural development policies, improvement of access to safe water and sanitation, and expansion of global partnership for development as small farmers are integrated in value chains. So on view of the all these studies in our India, so in India in 2016, northern states of Sikkim achieved its goal of converting to 100% organic farming. Other states of India, including Kerala, Mizoram, Goa, Rajasthan, and Meghalaya, have declared their intentions to shift to fully organic cultivation. The South India states, Andhra Pradesh is also promoting organic farming, especially zero budget natural farming, which is a form of regenerative agriculture. As of 2018, India has the largest number of organic farmers in the world and constitutes more than 30% of the organic farmers globally. India has 8.35 lakh certified organic producers. So conclusion of the presentation is there is no doubt that biofertilizers are the potential tools for sustainable agriculture, not only in India, but also globally. Our dependence on chemical fertilizers and pesticides has encouraged the thriving of industries that are producing life-threatening chemicals which are not only hazardous for human consumption, but also disturb the ecological balance. So biofertilizers being essential components of organic farming play a vital role in maintaining long-term soil fertility and sustainability by fixing atmospheric dinitrogen, mobilizing fixed macro and micronutrients, or convert insoluble phosphorus in the soil into forms available to plants, thereby increases their efficiency and availability. Okay. The application of biofertilizer can minimize the use of chemical fertilizer, decrease environmental hazards, enhance soil structure, and promote agriculture. So biofertilizer, no doubt, they are cheaper and remarkable in affecting the yield of cereal crops. Biofertilizer being important components of organic farming play a key role in maintaining long-term soil fertility and sustainability by fixing insoluble phosphorus in soil into forms available to plant, thus increasing the effectiveness and availability. In context, both the cost and environmental impacts of chemical fertilizer, excessive reliance on the chemical fertilizer is not a useful strategy in the long run. So these are the some references. Thank you. I am very grateful to the organizer giving me a chance to present here on a topic that is biofertilizer. Thank you, one and all.
Thank you so much, sir. Thank you so much. You have put light and focused on the very important topic, which is a buzzword, that is biofertilizer, a modern approach on the organic farming. You put light and focused on how the cultivating land and raising crops and to keep soil alive in good health, and by use of the organic as well as along with the other waste, that is um, aquatic waste, crop waste, and other biological materials, and to keep our soil alive and in good health. Thank you so much, sir. I take privilege to express my deep sense of gratitude towards you on behalf of all trustees, Amolak Jain Vidya Prasarak Mandal Kada, as well as on behalf of our dynamic principal, respected N.S. Rathi sir, as well as on behalf of Department of Botany, S.K. Gandhi College Kada. Once again, thank you for your eye-opener talk, sir. May I now request to enrich us with another important aspect by Professor Dr. B.D. Takte sir from Nevasa to discuss with us on this important topic of use of biofertilizer for our sustainable development. May I now request technical assistance to give Takte sir the spotlight, please. Sir, would you please start your video, Takte sir?
Hello.
Sir, please, may I request you to start your video, Takte sir? We cannot provide you the spotlight. Takte sir, please start your video. Yes, thank you. Thank you so much, sir.
Yes, ma'am.
Sir, we welcome you wholeheartedly. On the behalf of Department of Botany, S.K. Gandhi College Kada, as well as all the trustee members, and on behalf of our principal, Dr. N.S. Rathi sir, we welcome you. Welcome, sir. May I now request Jadwar sir, Professor Prakash Jadwar, to come forward to introduce Takte sir. Sir, please.
Hey, Santosh.
Thank you, madam. For this another session of our national webinar on role of biofertilizers for sustainable agriculture development, we are here with another speaker, that is Dr. B.D. Takte sir. His educational qualification is MSc PhD, and he is working as a scientist at Krishi Vigyan Farm at Bhenda, Taluka Nevasa, District Ahmednagar. He has worked as an assistant professor at Jijamata College, Bhenda from 2004 to 2007, and also he has worked as in charge of soil testing laboratory. He has attended training program on biofertilizers and quality control at Indian Agricultural Research Institute, New Delhi. He has also published 10 international papers in reputed journals, and his research area is isolation of local strain of azotobacter and its effect on Zea mays, and also his interested area is biofertilizer production. Now I request to Dr. Takte sir to please start his lecture.
Thank you so much, Jadwar sir, for giving nice introduction of Professor Takte sir. May I now request Takte sir to deliver his lecture, sir, time allotted to you. As we have some technical and time constraint, hope you will kindly cooperate for next 30 minutes. Thank you, sir. Sir, hope you will kindly unmute yourself. You are muted, sir. Sir, please unmute yourself. Takte sir. Hope you will kindly unmute yourself, sir. Hope you will kindly start your video and kindly unmute yourself, sir. Professor Takte, sir. Yes, sir. Thank you so much.
Hello.
Hello, sir. Yes, sir. Hello, sir. Takte, sir. You are audible, sir. Dear friends, hope all of you will keep open your videos and kindly keep muted your mics. Sir, you are requested to guide us on this important topic of use of biofertilizer for sustainable development. Sir, please.
कोणतं कॉपी करा त्याला ना आपल्या पाहिजे टाकायचं कॉपी
Takte, sir, we have spotlighted you, sir.
शेअर मध्ये फोल्डर कुठे आहे कॉपी आहे आता येत नाही घ्यायला भेटलं डायरेक्ट कशा बॅक ओपन असणार लिंक
As we know, with the encouragement, enthusiasm, and excellency, it is one of the major aspects of our organization, Gandhi College, Kada, with an object of encouraging the spirit of researchers and innovations. With this aim, Department of Botany feels proud to host this national webinar. This webinar and this event is not just a single day routine seminar, rather than it is a composition of 4 eminent speakers enlightening us on this occasion, each specialized in specific knowledge areas. We are now welcoming once again Akshay Takate, sir. I think he is getting some technical hitch which would be solved earlier. Takte, sir, please.
बॅकलोड का बरं
Yes, Takte, sir. Hello. Yes, Takte, sir, please. We have spotlighted you. Could you be able to please start your video? Akshay Takte, sir. Good afternoon and welcome, Akshay Takte, sir. Sir, could you please start your video? We have spotlighted you. We are eager to hear Akshay Takate, sir. Hope you will kindly start your video, sir. We welcome you wholeheartedly on behalf of SK Gandhi College, Kada, Department of Botany, to Akshay Takate, sir. Sir, your video is again off, sir, and you are muted. Yes, sir.
Yes.
Yes, sir. Hope you will kindly keep your camera in front of you. We are unable to see you, sir.
एक काम कर, इधर घे बरं कसं open करते, एक 2 मिनिट.
Yes, sir.
Yes.
Sir, we are unable to see you. Hope your camera will be tilted slightly. Yes, sir. Yes, sir. Now we can see you, sir. Yes, sir. You can proceed, please, sir. Good afternoon and welcome.
Okay, madam. Good afternoon. Yes. Hello, madam. Hello.
You are audible, sir. You are very well audible, sir. You can
Okay, madam.
continue.
Here in the today's one day webinar, I am thankful to the organizing secretary, then principal of the college, and respected Monari Chari sir, respected Ade sir, respected principal, Dr. Rathi sir, vice principal, Dr. Patali sir, and organizing committee of this webinar and Department of Botany. Today I am discuss about the biofertilizers for the sustainable agriculture. In the last century, the chemical fertilizers were used in the agriculture, and farmers were happy to getting in the beginning. But slowly the chemical fertilizers started displaying ill effects such as leaching out of this chemical, then polluting water, destroying microorganisms, making the crop more susceptible to the attack of diseases, and reducing the soil fertility, and thus causes irreparable damage to the overall ecosystem. Here, this is for overcome a solution. Now here, what are the biofertilizers? So the biofertilizers contain living organisms which, when applied to the soil, which colonize the hydrosphere while increasing the supply of availability of primary nutrients to the host plant. This biofertilizer adds nutrients throughout the natural process of the nitrogen fixation, solubilization of phosphate, and stimulating the plant growth through the synthesis of growth promoting substances. Here, for the crop growth, the different 16 nutrients are required, which are classified into the major nutrients, secondary nutrients, and micronutrients. So major nutrients are nitrogen, phosphate, and potassium. Secondary nutrients are calcium, magnesium, sulfur, and micronutrients are zinc, ferrous, copper, manganese, boron, molybdenum, silicon, then sodium, cobalt, and chlorine. So these nutrients which are supplied through the synthetic fertilizers, but these synthetic fertilizers causes ill effect on the soil health and which reduces the microflora from the soil. So if you consider here, the 1 teaspoon of the soil contains 1 million of the bacteria, then that soil we called it as a fertile soil. And when this microflora goes on deteriorating, the fertility of the soil goes on decreasing. Means there are different kinds of organisms which maintain physical, chemical and biological properties of the soil and which help to convert the insoluble fertilizers into the soluble form and that easily get available to the crop. So there are different microflora. There are many microbes, that is insects, then some bacteria, fungi, actinomycetes, some protozoa, which are present in the soil to maintain soil health. But due to agricultural practices, that is the application of heavy doses of chemical fertilizers, that is application of heavy doses of biological pesticides, then adding the pollutant in the water basin and the imbalanced doses of chemical fertilizers as well as heavy irrigation, which causes the irreparable damage to the soil. So there is a solution. The number of intellectuals throughout the world started working on the alternative and found that biofertilizer can help or increasing the yield without causing damage to the associated with the chemical fertilizer. And now, what is the biofertilizer? So it is a large population of a specific or a group of beneficial microorganisms for enhancing the productivity or by solubilizing soil phosphorus or mobilizing the potassium or by stimulating plant growth through the synthesis of growth promoting substances. The biofertilizer based on the renewable energy sources are cost effective, eco-friendly and can help to economize on the high investment needed for the chemical fertilizer. So there are different types of the fertilizers. These fertilizers were described by the Ade sir and I think Monali Chari sir. That is about the rhizobium and azotobacter. These types of these fertilizers. So according to their mode of action, which are classified into the nitrogen fixing bacteria, then phosphate solubilizing biofertilizers, which are classified into two groups, that is phosphate solubilizing bacteria and phosphate mobilizing bacteria, then potash mobilizing biofertilizers. Then there are certain organisms which help to soluble the insoluble micronutrients like zinc and ferrous, and some group of microorganisms that is the rhizobacteria. We called it as plant growth promoting rhizobacteria, Pseudomonas trichoderma. How the biofertilizer plays an important role. So biofertilizer helps to fix the atmospheric nitrogen. They are also producing some antibiotics, means which helps to give certain help to uptake certain nutrients as well as produces certain antibiotics. They are also producing growth promoting substances. They also stimulate the soil microorganisms and also enhance seed germination, acts as a biocontrol agent, also acts as a phosphate solubilizer, and to contribute about fixation of nitrogen, that is nitrogen present in our environment. Now the biofertilization and biocontrol, these both the terms which are important in biofertilizer, because PGPR is most plant growth promoting rhizobacteria, including azotobacter, acetobacter, azospirillum, then rhizobium, phosphate stabilizing bacteria. They can survive around the rhizosphere or in the rhizosphere around the root system of the plants, and which helps to fix the atmospheric nitrogen, siderophore production, phosphate stabilization, and for the control of certain diseases, that is antibiotics, then lytic enzyme production. Because of that, the yield and yield contributing traits of the crops were increased. Then second most important thing is that while considering the biofertilizers in our atmosphere, there is about 78% of the nitrogen. But this nitrogen plant cannot take itself. For the availability of this nitrogen, which comes on Earth surface with the help of lightning during the lightning in the cloud during rainy season, and with the help of certain microorganisms. But when we apply this synthetic nitrogen and consider the nitrogen cycle, so this rhizobacteria only trap the atmospheric nitrogen, and which get fixed around the root system of the plant. But conversion of this nitrate, because conversion of this ammonia, that is into the nitrate, because nitrate is the source of nitrogen that plant can accept. So the conversion of this nitrogen takes place only through the bacteria. That bacteria including nitrifying bacteria, and these are present in the compost fertilizers or in the vermicompost fertilizers. So for the conversion of this nitrogen, which is fixed by the bacteria into the nitrite form, that takes place through the nitrifying bacteria. So this is the nitrogen cycle. Means apply nitrogen, it may be in the form of synthetic fertilizer. The conversion of this nitrogen takes place only through the microorganism. Hence, the microorganism plays an important role in sustainable agriculture. That is the biofertilizer. So, as a consider here different nitrogen fixing bacteria, that is the azotobacter, that is described earlier. But here, azotobacter is a free living nitrogen fixing bacteria which lives around the root system of the plants and fix the atmospheric nitrogen for the non-leguminous crops, that is dicotyledon and cereal crop. This azotobacter is very better biofertilizer. But it is a drawback is that for the fixation of nitrogen in the soil, our soil should be rich with the organic carbon, because the activity of the azotobacter of fixation of nitrogen takes place through the absorption of organic carbon which is present in the soil. So while using this azotobacter in the agriculture for the crop production, we must introduce certain organic sources of the organic carbon. This organic matter or organic carbon should be more in the soil. Then and then this azotobacter will be survive in that soil and will help to trap the atmospheric nitrogen in the soil. So this is the effective organism. It produces certain indole acetic acid, then gibberellic acid, and which helps to enhance the root system of the plant, white root of the plant, and also help to control certain pathogens which are present around the root system of the plant. So that is the azotobacter. Now second most important bacteria, so that is called as acetobacter. So acetobacter, you know that rhizobium is a symbiotic nitrogen fixing bacteria. Rhizobium is a symbiotic nitrogen fixing bacteria. That rhizobium is also required for the leguminous crop, and which is root symbiont only. But acetobacter is very effective organism that lives around the in the root also, in the stem and in the leaves of the crop like sugarcane, because most of the sugarcane grower farmers, they should use this acetobacter. Acetobacter is whole plant symbiotic organism which is discovered earlier in Brazil by Dauvergne, and it found that it is very effective for the nitrogen fixation. But while using this bacteria, we have to give the seed treatment or set treatment to the sugarcane crop. We have to dip the state of this sugarcane crop in the solution of this or in the inoculum of the acetobacter for 30 minutes, and then you have to plant that set. It will help to enter the bacterium inside the set and will help to fix the atmospheric nitrogen throughout the life of that sugarcane crop. But while using this acetobacter, we have to reduce the 50% dose of recommended dose of nitrogen fertilizer. So while using this acetobacter, we have to analyze the soil sample first, and then after while application of this acetobacter, we have to reduce the 50% dose of the nitrogen. That will help to minimize the chemical fertilizer application of chemical fertilizers like urea, and will help to maintain the or will help to fix the atmospheric nitrogen throughout that crop. It is very effective method for the application of acetobacter. So acetobacter is very effective bacterium which is whole plant symbiotic organism. Now next one, that is the these are included in the nitrogen fixation, because you know that in our atmosphere nitrogen is about 78%. So it is very important to fix the atmospheric nitrogen. So we can reduce the cost of nitrogen of chemical fertilizers by using this bacterium like acetobacter, azospirillum, then rhizobium and azotobacter. So azotobacter is, rhizobium is crop specific. There are 7 different groups of this rhizobium. Rhizobium we can use only for the leguminous crop. Azotobacter we can use for monocotyledon crops as well as some dicotyledon crops, but we have to maintain organic carbon above 1.5 to 1%. Then and then it will be it will show you a very better result of the nitrogen fixation as well as for the soil health maintenance. Then azospirillum is also associative organism. It lives inside the some extent in the root system of the plant and in some extent in the soil also. So it will be it is an associative organism, and mostly it is effective for banana crop. We can use it for banana and other cereal crop, the azospirillum. But acetobacter is very effective for the nitrogen fixation of sugarcane crop. And for other crop, you can use the azotobacter. Now, second most important fertilizer, that is the phosphate. You know that this phosphate is a very effective nutrient required for the crop. But now when the soil, we analyze the soil, our soil is mostly the phosphate is fixed in the soil because of the salinity of the soil and pH of the soil. So phosphate availability of the soil, which is totally depend upon the pH of the soil. That is, if our pH is more than 7.5, then our the applied phosphate in the soil remains fixed. So for the conversion of this phosphate into the available form, the soil pH should be 6.5 to the 7.5. Then and then the availability of the phosphate will be better. But our soil is salinity of the soil and most of the soil become basic now, and that soil the phosphate in the soil gets fixed. So for the availability of this soil, the microorganism plays very important role. The phosphate solubilizing organism, that is phosphate solubilizing bacteria or phosphate solubilizing microorganism. So there are so many organisms which help to solubilize the insoluble phosphate. So bacteria like Bacillus megaterium, Bacillus phosphaticum, that's called, then Bacillus polymyxa, then Pseudomonas striata. So these are the bacterial species which help to solubilize the insoluble phosphate. And other some organisms also, that is Aspergillus avamori, Aspergillus niger also plays an important role in phosphate solubilization. Then next phosphate solubilizer that is earlier described by Monali sir, that availability that on the mycorrhiza. So mycorrhiza is also a very important phosphate solubilizing bacteria. So which fungi. This mycorrhiza grows on the roots of the plant and helps to convert the or helps to mobile the phosphate from soil to the crop, and also that helps to develop on the root system. So it protects the growth of pathogenic fungus around the root system. Means this helps to control the diseases as well as helps to convert the insoluble phosphate as well as insoluble micronutrients also transport helps to mobile these insoluble minerals or nutrients to the crop plant with the help of this mycorrhizal fertilizer. So these are the phosphate stabilizer. So second most important that nitrogen, then phosphate, and second most important nutrient that is the potassium. So in Maharashtra, the if you analyze the soil, this that is rich with the salinity of the soil, because our electrical conductivity of the soil is more than one now. Most of the soil, their electrical conductivity is more than one. So in such a condition, the availability of this nutrient become lower, and for the fixation of this nutrient becomes in large amount in the soil. So for the conversion of this insoluble nutrient into the soluble form, this microbes plays important role. Here in the mobilization of this potassium, because potassium gets very easily available to the crop through the irrigation water also. But due to the high salinity, this becomes fixed in the soil or becomes immobile in the soil. So for the conversion of this phosphate into the mobile form, so there are certain organisms, that is bacillus species and Faturi aurantia. These are the organisms plays important role to convert this insoluble potassium into the immobile potassium into the mobile form, and we get easily available to the crop plant. So that is these are the three major nutrients we can make the availability through using these biofertilizers. The next most important organism that our soil has some waste material in our agricultural land, but most of the farmers they burn this organic waste, and due to that it pollute the soil as well as pollute the our environment. So for conversion of this organic waste into the good compost, there are organisms are there that organisms easily decompose this organic matter are very better compost. We call it as a compost fertilizer. This compost fertilizer helps to maintain the organic soil. So this
Hello, sir. Hello, sir. Honorable Takte, sir. You have, by mistake, you have mute yourself, sir. You got muted, sir.
compost is possible through using the fungi, that is Aspergillus niger, Trichoderma viride, Penicillium chrysogenum, Chitomium globosum. So these are the some organisms. They help to convert this organic compost into a organic compost, decomposition of organic matter into the good compost, which helps to maintain the organic carbon in the soil.
Hello, sir. We are unable to hear. Hello. Hello, Akshay Takte, sir. Hello, sir. Professor Takte, sir. Could you hear me?
Okay. Thank you, sir.
We are unable to hear, sir. Sir, we are unable to hear, sir. We could not listen you. Hello, sir.
Okay. Hello. Hello.
Yes, sir. Yes, sir. Yes, sir. Yes, sir. You can continue.
Okay. Okay, madam. I am very thankful to the organizing committee of the college, principal, Dr. Rathi sir, Vice Principal sir, and all organizing committee. Thank you very much.
Thank you so much, sir, for your insightful
Hello.
Esteemed, hi, hello, sir. Thank you so much, sir,
Hello.
for your esteemed
Hello.
and insightful guidance. Hello, sir. Myself.
Thank you, madam. Thank you.
Yes, sir. I would like to thank you. I would like to express our gratitude towards you on behalf of all the trustees, Sri Amolak Jain Vidya Prasarak Mandal Kada, as well as on behalf of our dynamic principal, our motivator, Dr. N.S. Rathi, as well as the on behalf of Department of Botany, I would like to thank you wholeheartedly for your esteemed guidance. You have provided us many important insights on this alarming topic of successful use of biofertilizers for sustainable development. Once again, we are so much thank you, sir. May I now request?
Thank you.
May I now request? May I now request a dynamic personality, innovator, motivator, and our backbone, without whose support we cannot even stand, and we can never proceed ahead to. May I now request none other than our honorable Principal Dr. N.S. Rathi sir, S.K. Gandhi College, Kada, to deliver presidential address. Rathi sir, please.
Hello.
Yes, sir. Yes, sir. Yes, sir, Rathi sir. Sir, we are unable to see you, sir. Yes, sir.
Now?
No, sir. I think there is no focus on your face, sir. Could you please adjust yourself? Yes, sir. Yes, sir. Now we can see you. Yes, sir. Sir, once again, we are unable to see you, please, sir. Could you please adjust? Yes, thank you, sir. Thank you.
Oh, audible, ma'am?
Yes, sir. Sure, sir.
Okay. Now, the last part of this particular session is, that is the presidential address. I am not going to take much more time because already all the organizers and the committee members, they are having the angry, and with the angry purpose, I feel that you should not be angry with me. So good afternoon to all of you. Today, I am feeling very much delighted to see you as a participant in the national webinar on role of biofertilizer for sustainable agriculture development. Though the pandemic situation of COVID-19 has passed some challenges before, the humanity and the human beings find the new ways of maintaining the relationship. So here what is going on? We must thank the technology for reducing the gap and bringing close to all of us through such digital platform. The farmers are facing the various problems due to the change in the climate, and because of the change in climate, such as pests, insects, etc., these things are becoming a very obstacle for the growth of the crops. So the role of biofertilizer is unique importance for the sustainable agriculture development. Keeping the same view in the mind, the IQC of our college has adopted the nurturing the mother earth as one of the best practice for the NAC. And the college has adopted the 11 villages in which the consultancy to the farmers is provided regarding the things like the weather forecast, then soil health card, pH of the water and soil, high yielding seeds, and so on, with the help of the agriculture officers of the state government. And therefore, we have introduced the certificate course in the organic farming and vermiculture at graduational level. I am very happy about the department that the department has taken this topic for discussion because it includes not only the botany subject but where the farmers are on the border of Delhi demanding for the farmers justification. Means farmers should get the good income, and the government is also planning to develop the agriculture sector. During the pandemic COVID-19 situation, the approach of the society has become very hygienic, that is the organic itself. And therefore, during the pandemic situation, the government are taking the initiative steps by declaring the emergency fund 30,000 crores of rupee, 30,000 crores introduced for the agriculture sector and 1 lakh crore for agri infrastructure. And through this only sustainable development of agriculture will be possible. And I'm very happy that this botany department, they are not kept only the technical subject, whereas agriculture sector development, they have also considered, and that topic relates with the economics and commerce too. And with this, the one of the you are the resource person, he talked about the green revolution that is related with the agriculture sector. But I add about the green revolution, the white revolution is also important. Then sun revolution is important, and the blue revolution is also important. All these revolutions are related with the agriculture sector itself. So all these sectors are also relates with the science itself. May be the botany or maybe the zoology or maybe the physics itself. So sustainable development, when we talk about this, that it's related, the government has to take the initiative for this, the development purpose, and especially the sustainable development includes economic, social, and environmental development. And all these development, it should be related with the viable, it should be economic one, it should be a bearable one. Then only it will be considered the one of the good work, and the result of this such particular webinar will be come to a certain conclusion. The eminent personality and the inaugurator of today's webinar, Professor Mohan Chari sir from the Osmania University, Hyderabad, has spared his time with us. We are very much feeling the joyful for having you with us, sir, on this occasion. We are speechless for sharing your vast experience with us, and you have given a roadmap for the control of the diseases on the plant and organic farming with the parental approach, because at the end you were asking very cautiously about the advisor and all the faculties of this, and this shows that your parental approach, sir, we are really very grateful for you. I am also indebted to the Avinash Ade sir from Pune and Dr. B.D. Takte sir and Umesh Mogle for guiding us the a national webinar. And about the Mogle sir has especially stressed on the organic farming, and with his presentation I feel that really he is not from the only from the science faculty, but he also concentrate on the economy of the agriculture commercial part, and with the income itself he explained in detail. Though the organization madam has given him 30 minutes, but he is a teacher, and he has taken more than the 50 minutes. So, but by explaining all these details, he could touch all the particular points. So I express special thanks for all these faculties for guiding us in this national webinar. I also express my sincere thanks to honorable members of our society, Amalok Jain Vidya Prasarak Mandal Skada, for their support to conduct such webinars. Naturally, my thanks also goes to on the account of the botany and its faculty members, that is the Dr. Sayyid IG, who is heading the department, and also praise the Dr. Anarse PS, co-convener of the webinar. I must also thank to the Dr. Patale SS, organizing secretary of this national webinar. My best compliments are with the Dr. PB Jadwar sir, who is a joint secretary of this webinar, and also admire the presence of Mr. Amar Wagule for his involvement on this occasion. I must also glorify the efforts of coordinator uh, of NAC itself for promoting the various departments for such activities, and therefore I also lastly the person without whose the technical support all this will be not possible to complete this webinar the technically. So Dr. Hase Yasar, and my best compliments are also with all of you. And last but not least, I am thankful to the departmental staff, non-teaching staff, Gaude NP and Mr. Gunkar BD for helping to conduct this webinar, and because the gap of this will be, they have filled it, and therefore please take the care, and I am very much thankful to one and all of again. And today's the link between all of us, that was the ma'am has played a Suparna ma'am has played a very important role between the different the dignitaries and the participant. So and those who have joined this webinar, I'm also very much thankful to all of them to cope up and to have a good webinar, and you have listened very patiently. So once again, I'm very much thankful to all of you. Thank you.
Thank you. Thank you so much, sir. You have enlightened us. You have focused on the importance of blue revolution and white revolution along with the green revolution. Thank you so much, sir, for your eminent and esteemed guidance to all of us. Now it's time to acknowledge speakers. May I now request Sayyid IG sir to propose vote of thanks as head of the Department of Botany, SK Gandhi College, Sakada. Sayyid sir, please.
I am here to express the vote of thanks for national webinar on the role of biofertilizer for sustainable agricultural development. First of all, I express my heartfelt gratitude to the Honorable Sri Kantilalji Chanodia, President of Sri Amalok Jain Vidya Prasarak Mandal Kada. We are also thankful to Honorable Sri Gokuldasji Meher, Honorable Yogesh Kumarji Bhandari for encouraging us. My sincere thanks to Honorable Sri Hemant Kumarji Pokharna, Secretary of Sri Amalok Jain Vidya Prasarak Mandal Kada. We are also indebted to Honorable Dr. Umesh Kumarji Gandhi, Treasurer and Coordinator of our college. The man who supported us for the organizing this national webinar, none other than Principal of our college, Dr. N.S. Rathi sir. We are speechless for our guidance. We appreciate Dr. Bhandari JM, Vice Principal of our college, for this support. We applaud the eminent keynote speaker of this webinar, Professor Sri C. Manoharacharya from Usmania University, Hyderabad, for sharing his valuable time for us. We are very much thankful to Professor Ade AB from SP University, Pune, Dr. B.T. Takte and Professor Umesh Mogle for their valuable guidance. I also recorded my thank for my faculty member, Dr. Anarse P.S., co-convener of this webinar, Dr. Patale S.S., organizing secretary, Dr. Jadwar P.B., joint organizing secretary, and Mr. Amar Waghule for their cooperation to conduct their national webinar. The man who had taken effort to bring all of us together on this digital platform, Mr. Hase SR, we prize our involvement with us. The participant also deserve thanks for being with us from long time. Lastly, I am also thankful to all staff of my college, along with Mr. Gaude NP and Mr. Gonkar BD for taking efforts to organize this webinar. Once again, thank you very much to all. Thank you.
Thank you so much, sir. Thank you once again. Thank you so much to all of the eminent speakers. Thank you so much to audience who remained patient throughout and encourage the moral of the speakers by their enthusiasm. Thank you so much all for being our pillars of support. With the permission of President of today's webinar, Principal Dr. N.S. Rathi sir, I would like to conclude today's webinar over here. Thank you once again, one and all.